Hello, everyone. Good afternoon. We hope everyone is doing well. My name is Rosimara Barbosa Bispo. I am a PhD candidate in genetics and plant breeding, general coordinator of study group Ganho Genético, and the general coordinator of InterGen. It is a pleasure to have and share this moment with all of you. At this moment, we started III InterGen Series with the theme Artificial Intelligence in Plant Breeding, New Approach to the Challenge of Climate Change. This symposium is promoted in partnership with the Corteva AgriScience Company. In this third edition, his event provides an opportunity for professionals, experts, and students to discuss how advances in artificial intelligence have contributed to studies and advances in the field of genetics and plant breeding, as well as to mitigate the effects of climate change. In this sense, the topic is extremely important since it integrates different approaches such as phenotyping, genotyping, and machine learning with the objective to contribute to the interpretation of data for decision making in the face of rapid environmental change. Thank you for your time and for choosing to participate in your symposium. Before we start with the lectures, let's get to know a little more about InterGen and its representatives. Let's see a video about it.
The Brazilian territory has a large area, housing several biomes which directly impact the country's agriculture. The challenge of meeting the growing demand for food in the world has led to the advancement of research in genetics and plant breeding, which enables the increase in productivity and the adaptation of different cultures to different environments. For decades, many universities have been working on the development and the improvement of different species of agronomic importance in addition to training professionals in the area. Considering the current demand of agriculture, young researchers in genetics and plant breeding not only need the technical knowledge learned and developed at the universities, it is also necessary to go beyond the research itself and seek as much information as possible through the exchange of experiences with professionals and students from different Brazilian regions. Thinking about these needs, InterGen was born. InterGen is an extension group in genetics and plant breeding, aiming to bring graduate students closer to plant breeding in Brazil. In addition to this main objective, the group promotes science through annual symposiums and meetings to integrate the groups and to contact researchers in the field of plant breeding. The InterGen group is also part of a network called the Plant Sciences Symposia Series, promoted by Corteva AgriScience. In this project, organized by several institutions around the world, students are encouraged to organize scientific events that promote discussion between the academic community and industry. Currently, InterGen is made up of 7 groups from different Brazilian states. InterGen from the Universidade Federal de Goiás, Gen from the Universidade Federal de Lavras, and the Gen Melhor from the Universidade Federal de Viçosa, representing the state of Minas Gerais. GEMP from the Universidade Estadual Paulista, Júlio de Mesquita Filho, and GVENC from the Universidade de São Paulo. Ganho Genético of the Universidade Estadual do Norte Fluminense, Darcy Ribeiro, representing Rio de Janeiro, and NewGem of the Universidade Federal do Rio Grande do Sul. The different plant breeding groups that are a part of InterGen are always interacting through the participation and dissemination of various webinars, lectures, workshops, and other events that each group carries out, building a support network among young researchers. For the next few years, InterGen members will also have mentoring to develop their soft and hard skills, allowing for more effective leadership and contribution to the groups they belong to. If you are a student in the field of plant breeding and want to participate with InterGen, get in touch with your university's group and contact us. We are open to the entry of new groups. And for you, who are a researcher, come be our partner and help us to develop future breeders, strengthening plant breeding in Brazil.
Hello everyone, my name is Rosimara Barbosa Bispo. I am the General Coordinator of InterGen in the 2022 and 2023 Management. I will part of the study group Ganho Genético at the Universidade Estadual do Norte Fluminense Darcy Ribeiro in the state of Rio de Janeiro. As you already know, InterGen is made of 7 universities from different Brazilian regions, with 2 representatives from each group. So let's get to know the other members.
Hello everyone, my name is Jean Torres Leite. I'm a part of study group Ganho Genético, linked to the graduate program in Genetic and Plant Breeding at the Universidade Estadual do Norte Fluminense Darcy Ribeiro in the Rio de Janeiro state.
Hello everyone, my name is Givanildo Silva. I'm part of the group Roland Vinkovski, linked to the graduate program in Genetics and Plant Breeding in the Escola Superior de Agricultura Luiz Queiroz in the University of São Paulo.
Hello everyone, my name is Gabriel Salgado. I'm part of the study group Roland Vinkovski, linked to a graduate program Genetics and Plant Breeding, Escola Superior de Agricultura Luiz Queiroz, Universidade de São Paulo.
Hello everyone, my name is Jaqueline Flores Schmitz. I'm part of study group NUGEN, linked to the graduate program in Plant Science Crop Breeding at Universidade Federal do Rio Grande do Sul from the state of Rio Grande do Sul.
Hello everyone, my name is Jessica Argenta. I'm part of study group NUGEN, linked to the graduate program in Plant Science Crop Breeding at Universidade Federal do Rio Grande do Sul from the state of Rio Grande do Sul.
Hi everyone, my name is Bruna de Figueiredo Duarte. I'm part of the study group J Melhor, linked to the graduate program in Genetics and Plant Breeding at the Universidade Federal de Viçosa in the state of Minas Gerais.
Hello everyone, my name is João Marcos Soares Ferreira. I am part of the J Melhor study group in Genetics and Plant Breeding at the Federal University of Viçosa in the states of Minas Gerais.
Hello everyone, my name is Leonardo Oliveira Silva da Costa. I'm part of the study group GEM, linked to the graduate program in Genetics and Plant Breeding, Universidade Federal de Lavras, Minas Gerais.
Hello everyone, my name is Mariana Gabriela Coelho de Queiroz. I'm part of the study group GEM, linked to the graduate program in Genetics and Plant Breeding, Universidade Federal de Lavras, Minas Gerais.
Hello everyone, my name is Luiz Gabriel Silva Varenga and I'm part of the study group EUGEN, linked to the graduate program in Genetics, Plant Breeding and Conservation of Plants from Universidade Federal de Goiás in the state of Goiás.
Hi everyone, my name is Giovanna Rodrigues Neves. I am part of EUGEN group, linked to Genetic and Plant Breeding graduated group at Universidade Federal de Goiás in the state of Goiás.
Great, beautiful people, beautiful videos, beautiful groups. So, continue the event. I invited Dr. Jason Hauscher from Corteva Agriscience, who will present the program Corteva Symposium Series. Dr. Jason is a passionate science and education advocate, leveraging the over 20 years of experience in academia, research, secondary education, and industry to foster mutually beneficial interaction between academia and the private sector in the agriculture and the biological sciences. Currently responsible for managing the Corteva Symposium Series with support a network of graduate students, organizes symposium at over 30 university and research centers around the world each year. Responsibilities also include the management of the New Frontiers in Applied Science Conference Series and support of the Corteva Internship Program for undergraduate and graduate interns working within R&D. Thank you, Dr. Jason. The audience is yours.
Thank you so much, Josimara. Can you hear me?
Yes, very good. And can you share your screen now?
Yep. Thank you for your patience. My computer seems to be slow this morning, but we will do our best. There we go.
No problem.
And hit one more button. Share. So let me know when you see the slides.
Yes, Jason, perfect. I can see your presentation and you can start. Jason, please, your microphone is turned off. Your microphone is off. Turn it on. Okay.
Okay, can you hear me again?
Oh, yes, now yes.
All right, I'll keep an eye on it.
Okay.
We're having strange computer issues this morning. So again, thank you for having me with you, and stop me if my sound disappears. So as Josimara mentioned, I am the R&D Academic Relations Manager at Corteva Agriscience, and one of the amazing things that I get to do as part of my job is to manage the Corteva Symposia series. So I'd like to take a few minutes to talk to you about our program. For those of you who are not familiar with it, and I know many of you are, but this is a global program where we support students to do just what you're seeing today, put together a scientific conference and use this opportunity to work together and develop skills that otherwise we think maybe don't have the chance to develop quite as much in normal graduate education programs. So this is a great opportunity, and we see this in 4 different categories. The learning that goes on, and I'm always fascinated by the particular speakers that students invite to these events. They get to curate their own scientific conference, which is very unique. Sometimes, you get to go to other scientific conferences, but you don't have control over who the speakers are. And in this case, the students do. So it's always great to see who they invite. It's great to build the networks. And there's no better example of this than InterGen, right? InterGen is the ultimate example of networking across Brazil, leveraging these interactions between universities and also to reach out to the world, which is what they're doing today. Career development, this is a great opportunity for students to get exposure to different possible careers in the future. Obviously, I work for a private company, Corteva Agriscience, and students should see what opportunities are out there in the private sector, as well as the public sector in places like universities and Embrapa, and so many opportunities out there. So this is a great chance for students to interact with scientists from every different possible path in the future. And then finally, soft skills. I'm always amazed at our Brazilian students who are so good at interacting and presenting these symposia, and they do such a good job, and they really exemplify the idea of teamwork, which is so important for any institution that's hiring future scientists, knowing that they can work on teams, work well together, collaborate. That is probably one of the most important skills that we look for in our future workers and scientists. So it's amazing to see here for InterGen. Let me see if I can advance my slides. There we go. So as I mentioned, this is a global event, and in this case, we are leveraging multiple institutions across Brazil, as that great video showed. InterGen is is a fantastic example of how this international global network is building communication, building interactions between institutions across the globe. And the scientific community, not only plant breeding, but the plant sciences and beyond, the agricultural sciences in general will benefit for this into the future. InterGen, as I've already said, InterGen plays a very special role. This is an institution that students showed the leadership to create. They saw the value in having these connections between different universities and different plant breeding programs. InterGen is, I think, the ultimate example, the most important example of how these kinds of programs can benefit students and how students can benefit the international scientific community. So again, thank you so much to this as a group. It's so great to meet with these students. I get to interact with them both at for the InterGen and at their individual institutions, which host symposia as well. So it's a great pleasure for me to work with them. And again, I just want to reflect on some of the history of this event over the last year. Obviously, we are in a transition year. 2021 was completely virtual because of the pandemic. 2022, we are starting to move back into in-person, but we've learned how valuable these virtual events can be. And continuing these through InterGen, I think is such a great opportunity to combine with the in-person events that we have across Brazil, to have this purely virtual event, which is really available to the community. As I mentioned, agriculture is not just plant breeding. It's not just plant sciences. It is broader than that. And so the symposia series is beginning to grow as well. So we've had a chemistry symposium, and we're hoping to have another one as well, probably next year, if not more than one. So we're always looking for new and interesting ways to grow and have the agricultural community interact. I've shown this example before, but I always love to talk about it. Our Brazilian students are always so creative. We had last year, we had a great event at the Brazilian Seed Technology Conference. They had a competition between institutions all across Brazil. So again, another great way for students to leverage the symposia series towards their education and towards the advancement of science in their particular disciplines. At Corteva Agriscience, diversity is really a very important part of how we see science moving forward. We cannot have innovative and science and agriculture in the future that drives sustainability if we do not have a diversity of people interacting. And so we have reached out with the symposia series to develop symposia in the United States at historically Black colleges and universities. They're called HBCUs. So we've reached out to those institutions to encourage a diversity of people to engage in agriculture and plant sciences. We have coming up the next event. You're welcome to go to our webpage to see our calendar, but University of São Paulo, it will have an event in October that I'm looking forward to. So please join us for that event. I also wanted to point out a couple of programs that we run. As I mentioned, diversity is so important to us. And so we have a program specifically dedicated to advancing students in underrepresented groups in science in the United States, right, from that perspective, and globally as well, to some extent. But this is a program where we have really been advancing that diversity in our company and in the scientific community. I do want to point out the availability of internships. I know in Brazil as well as the United States, there are internships as well. You can look for internships on our careers page, and you can filter by country to look for those. I'm not as familiar with how they work in Brazil, but in the United States, we have them. The advertisements come out this time of year through September, and you can look for available internships there. And another opportunity to interact with Corteva is the Open Innovation Platform. So this is a part of Corteva where we invite researchers from around the world to innovate with us. So if you have an idea of a project where we could collaborate with you, this is a great opportunity to leverage the strengths that a large company like Corteva has and your ideas, right? So we can make your ideas happen. We also have challenges that we set out, that we are looking for partners in the academic community to help us answer some of these questions, or to come up with proposals to accept these challenges and come up with solutions to them. And then finally, I wanted to talk about a conference that's coming up in a couple of weeks at Corteva. New Frontiers is a conference series that we support every year. And this year it is dedicated to sustainable agriculture. This will be a free webinar available to everyone. If you are interested in that, please reach out to me. I also have a LinkedIn post on it where you can, if you go to my LinkedIn page, you'll be able to find the post and for registration for this event. So students, faculty, you're all invited to participate. We have some great speakers lined up. It's actually a 2-day event. On the 11th, it's going to be dedicated to cropping systems for sustainable agriculture, everything from soils to new potential crops to livestock. So a very diverse lineup. And then on the October 13th, we have a second day of this event where it's really going to be focused on delivering crop protection solutions to farmers in the field. And how do we do that in a sustainable way? How do we target crop protection products in ways that don't impact the environment? So I think it's going to be a great 2 days of talks. So I invite you to join us for those. And to do that, please reach out to me, connect to me on LinkedIn. Here you can use the QR code there to connect to me. And it would be a pleasure to have you join us for that event and for all of our upcoming symposia. So I am going to stop sharing now and hand it back to Josimara so we can continue with the great symposium today. I'm looking forward to some great talks, and I really appreciate being able to join you.
Okay, thank you very much, Dr. Jason. On behalf of InterGen, I thank you, Corteva, for supporting the event. And with years, we learn more and have new experience. This is very important for the group's growth. Thank you so much. So, continue the event. We'll start the lecture of the first day with the subject Computational Intelligence, which algorithms have contributed to the improvement in agricultural yields. We invited Dr. Auti, professor and researcher of Wagner University. Dr. Auti did his PhD in structural biology at Utrecht University, focused on computational methods for protein docking. Subsequently, he worked for several years as a researcher in bioinformatics at the Wagner Plant Research. In 2016, he started as an assistant professor in the bioinformatics group at the Wagner University, where he currently is an associate professor. His research involves the development and the application of machine learning approaches to analyze plant omics data towards modeling of networks and computational analysis of protein and DNA sequence and structure. Thank you so much, Dr. Auti, for accepting our invitation. It is an honor to have you here with us. So before I give the floor to you, I would like to inform participants that questions and doubts about the lectures can be asked through the YouTube channel chat. The question will be answered by the speakers at the round table at the last talk of the day. So, Dr. Alt, you have 40 to 50 minutes to make your presentation, and the floor is yours.
All right, let me see if I can share my screen.
Okay.
Is this working?
Yes.
Seems to me that it's working, right? Okay,
Yes. All right. All right.
then I think I'm good to go. So yeah, I would like to start by thanking you for the invitation. I think it's really an impressive symposium to be part of, and I really enjoyed the overview that was just given by Jason. So it's really my pleasure to be talking here today about the application of machine learning on plant omics data. I had originally planned to give this lecture from Wageningen University. So this is at the left-hand side. You see the building where one of the buildings on our campus. Unfortunately, I didn't feel completely well the last few days. So I'm currently at home in my hometown Utrecht. And here in the second picture from the left, you see the Dom Tower that my city is famous for. So if you ever visit us in Wageningen, also be sure to have a look at Utrecht in the middle of the Netherlands. Yeah, and one unfortunate thing is that I will not be able to join in the round table for that reason. So, but I'm still happy to be giving this lecture here today. I want to start by acknowledging a couple of people, some of my collaborators from Wageningen University, but in particular the three PhDs that did some of the work that I will show in this lecture. The idea of this lecture is that I will give some general overview of how machine learning is applied on plant omics data, but I will use some specific examples from my own group to give a bit more detail. And that is work done by Janani, Sefking, and Muhammad. All right, so let me also briefly talk about my background. I think it was already nicely introduced, but just to give a bit more, to briefly summarize this, I did my PhD in structural biology, and you will see at some moment in this talk that now is also a very good time to think of using protein structures in machine learning, but I will get back to that later. And then currently I would maybe use the phrase plant system biology to summarize my my research, but this really is mainly using machine learning on plant data. A lot of it is on Arabidopsis, but we're also moving more towards applications in various crop species. And here on the map, I'm showing where I'm actually located. So this small dot here is Wageningen, and here again is one of our buildings. So let me move to kind of an overview of what this talk is about. And that is that when we talk about plants and omics data, we have really a lot of data at various scales available these days. So there have been a lot of improvements in measurement technologies, which result in the availability of data at various scales. And this is a picture that I took from a review that we wrote last year. I have references to I have a reference list at the end of this set of slides, so this you can maybe look at later if you want. But so this picture visualizes this data that we have at many different scales. So it ranges from the genotype and the genome level via biochemical levels of transcriptomics and metabolomics to data at the scale of plants or even larger scales. And the thing is that if we want to learn from the data, we will need to use computation to somehow connect the data and try to understand the meaning of the data. And there are, of course, different ways to do that. And very broadly speaking, you could think that on the one hand, you can use more mechanistic approaches to try and model data, and that is great if you can do it. But the point is that for a lot of this data, for a lot of these systems, we don't have sufficient understanding to make mechanistic models. And then very often machine learning can be useful, because what machine learning involves is finding patterns in data, and these patterns you can then also use to make connections between different types of data. So I would say that given the great increase in the amount of available data, and then the need to integrate and connect this data, that is one of the key reasons why machine learning is getting a lot of attention these days, in order to make sense of this type of data. Now in this picture I give a bit more detail about these different data types. I will not discuss them now in detail. I think many of you have a lot of knowledge about this. So we can think of data using sequencing, we think of data obtained using mass spectrometry. But this visualizes a bit the type of omics data that we typically work with when we apply machine learning on plant omics data. And here at the bottom you see kind of an overview of how you can maybe categorize the different applications of machine learning on this type of data. And this is also a little bit the flow of this lecture today. So I will talk first about using machine learning more at the level of genomic regions. So can we understand or predict properties of genome regions? And then I will talk about other type of machine learning where we more focus on genes and how these are regulated or what their functions are. And after that, I will also talk about some more recent, very interesting developments in new developments in the use of machine learning on plant omics data. All right, so we first start now on the use of machine learning to analyze or predict properties of genome regions. And you can think here of various things that you might want to do. Some of these are very classical tasks, like annotating genomes, and people have done that in various ways. But one thing that you these days really can do is to use machine learning to annotate genomes. And what that would involve is that you need to have examples, because this is a typical thing with machine learning. You need examples of what you want to predict, and then you can learn to predict new things. So that would be one thing on the level of genome regions. You can also think of predicting which genes are active versus pseudogenes. There are applications at the population genetic level. I show a picture here of what that looks like. So it involves predicting using, for example, resequencing data from a population, predicting where selective sweeps took place. But I want to give a specific example from my own research, which involves the prediction of crossovers. So we'll talk a bit more in detail to use that as an example of how we can do machine learning at the genome region level. So, the idea here is that we want to predict crossovers. So this involves the regions in the DNA where the paternal and the maternal DNA, where you have a switch between paternal and maternal DNA. So I visualize here very schematically how that happens during meiosis. So in blue and red we have the paternal and maternal chromosomes, and then well, I'm not going into detail here, but the effect of the meiosis is that you get this mixing and matching between these two types of DNA. And this is, of course, a very essential process. It is of key relevance for evolution, and it generates diversity. So it's very interesting to study from a more fundamental perspective, but also for breeding application, of course, crossovers are key. So there's been quite a lot of studies where people experimentally investigate where crossovers take place, and the point here is that these crossovers don't take place at random positions in the genome, but you can really find certain patterns. At the same time, we don't understand sufficiently well how these patterns are generated, or what exactly determines the preference for crossovers to occur in a certain location, not in another location. So for these reasons, we thought it would be could be very useful to try and use machine learning to predict crossover locations. And I'm showing here two examples of training data that we had available. So these are large-scale data sets where people in a certain, based on a certain cross, they investigated the occurrence of crossover. So here we have the different chromosomes, for example, of Abruptus or tomato, and then the bars here indicate the number of crossovers. But underlying that is that we also know the exact locations of these crossovers, or to a certain extent we know their exact location. I don't show it here, but this gives an idea of the data that we have available. So our goal was then to use machine learning to predict where in the genome these crossovers are likely to occur. And I would like first now to consider why this is a machine learning, how you can frame this as a machine learning problem. And in particular, we framed it as a so-called classification problem. So classification means that we try and predict multiple different classes based on some properties of the data. So classification is one of the main examples of, or main tasks in machine learning. And here at the top left, I visualize in a very schematic way what classification is about. So here we have red and black circles, and in this very simple classification example, our task would be to predict whether a circle is black or red based on the position of these circles, so based on x1 or x2. And x1 and x2 would be the position. But in our DNA-based model, it would be some numbers that somehow represent the DNA. And I will talk in a minute about how we can do that. But this is the technical term here is features, and it's good to realize that, of course, here in this simple example, we just have two features, but in realistic examples, you would have many of these features. You would have many numbers that somehow characterize your input data. And so to map this example, this toy example, to the crossover prediction, you need to think that maybe the black circles are locations in the genome where we experimentally found crossover, and then the red would be positions in the DNA where experimentally we did not find crossovers. This last point is a little bit tricky because, of course, if there was no crossover measured, it doesn't mean that the crossover cannot occur there. So we did some work to decide how exactly to sample these negative regions, but I will not talk about it now in detail. You can read it in the paper if you're interested. And this is actually also an example of something that happens quite often in this type of machine learning on omics data, is that you maybe have a set of positive measurements, but for your negative measurements, so the examples from the other class, might be more difficult to get these. Anyway, I talked about how we can frame the crossover prediction, so predicting properties of genomic region, in this case, whether they are crossover sites or not, how we can frame that as a classification problem. And now I want to explain how we can indeed convert genome sequence to numeric input, so these so-called features. And there are different ways that you can think of. In some approaches, people maybe count the occurrence of certain k-mers, and you can think of many different things. In our case, we used a combination of multiple things. We looked at sequence variation, and then the absence or presence of certain variation would be a number. We looked at the occurrence of transposons in the DNA, and again, the absence or presence of certain transposons would be a number. We also looked at the occurrence of dinucleotides, so this again gives you a number, how much of these dinucleotides occur or not in a certain genome region. And we also use an approach which predicts the kind of the three-dimensional structure of the DNA, and that again is a set of numbers that we can use to characterize that a given genome region. So then we use this training data that I showed before, and on this slide I summarize a bit what the model performance was. I can imagine that not all of you are familiar with this kind of plot, so let me briefly talk about it. We have here a visualization of the false positive rate and the true positive rate. These are two different numbers that tell you how good is the model. And then depending on some cut-offs in the model, we get different false positive rate and different true positive rate. So this curve characterizes the performance of the whole model. And the more to the top left this curve is, the better the prediction. So these three different colors here indicate three different machine learning models. A decision tree in green, a logistic regression model, and a random forest. And you can see here that the random forest model worked best in this case. Now, in addition to the prediction itself, which has quite decent performance, I would say, but in addition, we also looked at how does the model make its prediction. And this is one key thing that I want to stress here today, is that in some cases, when people use machine learning, they are maybe not so interested in why does the models make certain prediction. Maybe you only want to predict something. That can be the case. But I think in the application of machine learning to omics data in plants, you would often also try and use the model to learn more about your system. And one way in which you can do that is to, like we show here at the right hand side, is to look a bit at which features are really important for the model to make the prediction. And I think if you have time, we will not discuss this now in detail, how does this work, but what the plot shows is like the top most important feature. So this gives you some feeling about what the model really found important. And in particular at the top, at the left-hand side, there's propeller twist and helical twist, and these are two aspects related to the DNA structure. And that was quite an interesting finding, that somehow the model thought that the predicted DNA structure was really important for it to make the predictions. So I want to spend a little bit more time on this topic of the model interpretation, because as I said, I think it's often quite important in our field. And here I want to take it a bit to the extreme, in the sense that I want to stress that some machine learning models, by their very nature, are quite well open for interpretation. And I show an example here at the right hand side. This is a so-called decision tree. And this is a very simplistic example, of course, but this decision tree starts here at the top with the question, and then you can say yes or no, and then you follow the decisions, and then you get the answer. And so here the model is completely transparent. You don't need to do any effort. If you have trained this model on some data, then subsequently you can also understand how it makes its predictions. At the left-hand side, it's very different. We have a neural network, or so-called deep learning model, and there we have several layers, and can in reality be much more complex than what I show here. And there it's much more difficult to understand how the model makes its predictions. You can still do effort to get it, but the point is that there are differences between how easily you can interpret model behavior. And of course, it's also good to consider why would we want to interpret a model. And I listed some reasons here, and I think that in our field, it's particularly relevant that model interpretation can help us to understand better our problem, because we can learn something about the system that we study. And also it can help to really define actions based on the model, and that would in our case involve for example, designing new experiments. And maybe for the crossover model that's not so clear, but I will soon talk about another example where you can indeed see that based on model interpretation you can also think of specific experiments that you can design. And actually this is kind of a summary of what I just said. So at the level of genome regions, the example with crossovers, there we can interpret the model and then think about what determines the position of crossovers. And actually but this is a bit more in the future, you can also think of how you can use that knowledge for modifications in order to maybe engineer to get crossovers in positions that in a breeding scheme you normally cannot get them, something like that. I do think that an example that I will talk about in a few minutes, which is at the level of proteins or genes, there it's much even more clear that indeed model interpretation can help you to make new experiments, because if you think of protein function prediction, then if your model somehow tells you which residues are important for that function, then that obviously can allow you to make, for example, use mutagenesis experiment to understand more about, in this case, enzyme specificity. All right, I think it's indeed a good moment then to move forward to this second type of applications of machine learning. So we move now from the level of genome regions to the level of genes or gene products, so proteins. And here again, there's a variety of things that people do. You can think of, for example, predicting expression levels of genes based on promoter sequences. And one quite interesting example that I really would suggest you can study if you're interested is from this paper from a few years ago, where people made a couple of different models to predict expression levels. And in particular, what I found interesting there is that they used, that they combined data from multiple different plant species. I think included maybe Arabidopsis, maize, and maybe other species as well. I don't remember exactly, but I think this kind of using information from multiple different species and trying to learn patterns across species boundaries is something that is still quite at an initial stage in the application of machine learning to plant omics data. So I do think this kind of work is very promising. But okay, this is one example of work at the level of genes. You can also think of prediction of gene regulatory networks, and there are a lot of examples where people do that. So using maybe expression data and then predicting which genes regulate which other genes. I will not talk about this now, but this is really a big field, and it's obviously it's obviously quite important and useful if you can indeed predict regulatory networks. You can also think of predicting, this is a bit of a specific example, but you can think of using expression levels of genes to predict, for example, traits of plants, maybe stress response of other traits. QTL candidate gene prioritization is also something that people do with machine learning. We did also some work in that direction. But I would like to briefly give a bit more detail about an example that I already briefly alluded to, which is the prediction of enzyme function. And this is work that we recently did on plant sesquiterpene synthesis. And I want here to discuss the most simple version of this predictor. We have also a more complicated version where we predict at more detail, but for this talk I will focus on the kind of predicting the first step in the production of sesquiterpenes, which is catalyzed by these enzymes. So they start, the sesquiterpene synthesis all start from FAP, and then they go through multiple different steps. But you can kind of categorize them based on the initial cyclization that takes place. So we have we have 6 different groups, basically. So this initial predictor focuses on predicting those classes. And what you need to know is that a typical plant has maybe dozens or even hundreds of different sesquiterpene syntheses, and then they all use the same substrate, but they all make different products. And these compounds are relevant for fragrance and other things, also defense sometimes, and they are also very useful in biotechnology applications. Well, I will not talk more about it now, but it's really there are different applications. But I want to talk here about the machine learning, and one thing that you can of course wonder is, do we need machine learning? Because maybe if we just look at sequence similarity of these enzymes, maybe that already tells us enough. But in this phylogeny of a set of characterized plant sesquiterpene synthase, you can see that phylogeny itself does not give sufficient information about function for these proteins. Because you can, so the different colors here indicate these 6 different groups that I just mentioned. And then you can see here examples where enzymes with the same color, so here's something in red and here's also something in red, they're very far apart in the phylogeny. And also you can see examples here at the top where we have all different colors very close to each other. So these are very similar sequences, but the enzymes still have different functions. So this visualizes that the prediction of the product that these enzymes make, you cannot do it by just making use of overall sequence similarity. Now we again frame this as a classification problem, like in the crossover example. And then, of course, we again needed these features that I talked about before. So we somehow needed to convert the enzyme information to some numbers that our models can use. And in this case, we decided to use both sequence and also structure. Now, I will not talk much about how that works, making these features, but yeah, you can imagine that, like in the previous example, we can convert sequences to numerical representations, but then in addition, we also generated structures for these, modeled structures for these proteins, and we converted these to features as well. And I will say something, if I have time, I will say something about using structures in a minute in some other context. For now, I want to focus on the result of this exercise, and it was quite interesting to observe that indeed adding structure features compared to using only sequence features, it really improved the model, but that also depended on how we analyze the performance of the model. So let me guide you through this table. We have here 3 different prediction approaches. One is based on overall sequence identity, so not really using machine learning. One is using machine learning with only sequence features, and then one is with machine learning including structure features. And in the lines here, we have 3 different ways by which we looked at the performance of the models. One is by randomly splitting the data without making any effort to do anything else. And this is something that in some applications of machine learning would be sufficient, would give you a good idea of the performance of your models. But we realized that in this case, we needed to do a bit more thinking, because we realized that if you just randomly split the sequences, so 80% to train your model and 20% to test it, then what you will get is that the model will, even based on sequence identity, will seem to work quite well, but that is primarily because there's a big bias in which sequences have been characterized. So there are many related enzymes from very closely related species that have been analyzed, and although these are not always the same function, you also can see that still quite often they do. So by just, especially if they're very, very, very related, so by just splitting randomly a training and a test set, then the performance is really not very indicative of a practical application of this model, because in practical application, you would be more interested in very novel enzymes where you don't have very similar sequences. So we also made a performance assessment by using some genus to train the model and other genus to characterize the performance, and also by separately using only unicots to train the model and then monocots to characterize the performance of the model. And you can see that in particular in the last case, which is really the most difficult kind of case, you see that using the machine learning model gives a much better performance. So the number here is the accuracy. So you can interpret this like 75% is correct, and here it's only 50% with using the sequence identity. So here we really see the importance of using structure features, and also that, of course, it depends on how you analyze your model performance. So this, I think, is good to keep in mind in general, actually. I would like to go briefly back to the point of model interpretation that I mentioned already. And because also this kind of model, where we have features at the sequence and structure level, we can really try and use process that tell us which parts of the protein are most important according to the model. And I'm showing some visualization here. Maybe we can ignore the right hand side, but let's focus on the left hand side. So this is an example of a protein structure from one of the plant sesquiterpene synthetases, and then the colors here indicate which parts of the protein are important according to the model. You can see that many of these positions are close to the active site, which is here, and that makes sense, of course. But there are also some positions somewhat further away. But one very interesting opportunity, and this is something that we're currently working on, is that we can now use these positions to also not only validate the overall prediction performance of the model, that was on this previous slide, but to also look at, okay, if we now have these predicted positions, can we maybe change the function of some of these enzymes? So we're currently designing some test cases for that, and yeah, I cannot tell you the results yet, but maybe another time. The point here, though, is that here again you see an example of why model interpretation can be really useful. So I'd like to move on now. We talked so far about two types of applications of two very broad types of applications of machine learning on plant omics data, namely using machine learning at the level of genome regions and using machine learning at the level of genes or gene products. And something goes wrong here. I'm sorry. I would now. Sorry, am I still? I'm not sure what's happening here. Okay, yeah, I'm back. Somehow something went wrong with my slides. I'm sorry, but I think I'm back now. So what I would like to do now in the next maybe 5 or 10 minutes or so is to talk about more recent developments in the application of machine learning on plant omics data. Because I think there are some very exciting developments. They're also connected with technical developments in machine learning in general. So let me guide you a bit through that. And the first thing I want to mention here is that there's currently a big interest in connecting machine learning approaches with mechanistic models. I mentioned at the beginning of this lecture that we often use machine learning because we want to maybe integrate or connect data and learn from our data. But we cannot do this with mechanistic models often because often we don't have them. But of course, there are also cases where we do have mechanistic models, maybe like models based on differential equations, for example, could be other things. But so if you have that kind of models already, but you also have much more data that does not fit so well with this type of mechanistic models, then it can be very interesting to think about combining machine learning with mechanistic models. And there are some different ways that people call this, and in particular I want to mention here this term physics-inspired machine learning or physics-inspired neural networks, and the picture here kind of visualizes that. This might be a bit scary if you don't like equations, but what it kind of visualizes is that the kind of neural network that is shown here at the left-hand side can also be combined with differential equation model that expresses some knowledge that you have about your system, and in particular you can combine that knowledge from a differential equation model. You can combine it in the loss function that you already define also for your neural network, so you can optimize these both things simultaneously. So you will then get a solution of your problem that is a good fit according to a neural network, but that also is in line with your mechanistic model. This type of approach is quite novel, and I don't think there's much examples of using it at plant level yet, but I'm sure this will change soon, and we're also ourselves working in that direction. So in particular, we are part of a new project that should start early next year, where we will apply this type of combination, this type of hybrid machine learning to predict stress resilience. And one example of the type of work that we will do there is visualized here in this slide, where we will make different modules to some machine learning modules that characterize aspects of plant behavior, and then we will combine that with existing models that tell you something about flowering time. And this we will initially develop in our blob, but in a later stage we will also work on this in a couple of different crop species. And I think this is really a very interesting development, this combination of machine learning with mechanistic models. A second recent development, well, it's not recent in the sense that, so a second recent development is the use of protein structures. Now, of course, protein structures are already available for quite some time, so people have also done machine learning on protein structures for a long time. But what really has changed very recently is that we can use much more protein structures as input, and this relates to the fact that there's this new AlphaFold prediction. So you can find at the EBI website, for example, you can find the predicted structures of millions of proteins. For example, for all of the proteins that are available in UniProt, you can now get a predicted structure of quite good quality in most cases. So this really opens up new opportunities of using protein structures as input for machine learning. I will not talk about examples here, but if you think back of the example that I just discussed on enzyme function, you can imagine that for that type of work, but also for many other applications of machine learning on omics data, using structures can be relevant. You can think of predicting of interactions, for example, and many other things. On this slide, I just want to visualize something more at the methodological level that we are working on ourselves in my group, which is that there's also really a need to work more on how can we represent structures as input. So this relates back to this point of the features that you make for your input cases. And there are really different ways that you can think of how to represent a protein as input for machine learning. And it might have really influence on your performance. But okay, let's move on. So the third recent development that I want to briefly discuss is that is so-called unsupervised analysis. The machine learning that I've talked so far about is mostly what we call supervised. Supervised means we have some example cases, for example, some proteins for which we know the function, and then we use those known cases to predict the function, to make a model that can predict the function of other cases. But you can also think of unsupervised analysis, where you don't want to predict a specific label of your cases, but you want to find more general patterns that somehow tell you something about your data. And examples would be clustering, for example, but also many other unsupervised analysis approaches exist. And actually, these are in a very active development these days. And one area in which you can find really interesting applications of unsupervised analysis is single-cell RNA sequencing. I think if you have time, I will not discuss now how single-cell sequencing works. Just what you need to know for the next few slides, it's single-cell sequencing means you get, in principle, expression levels of many different cells for many different genes, for many different cells that you collected. And I want to briefly explain a little bit about what this unsupervised analysis of this type of data can involve. And in particular, I want to contrast it with the use of PCA, principal component analysis, that I think many of you will know about it. PCA is great, and you should definitely try it in general if you think it can help you. But it doesn't always work, and that relates to the fact that it's a linear approach. And so if there's patterns in your data that are not linear, then you will not find it. And also, the thing is that PCA really focuses on, let's say, the global structure in your data. So there have been quite active developments in alternative approaches that more focus on local structure. And one main idea in these approaches is that you will look at the distances between different data points, and then you try to find a way to represent your data in such a way that these distances, these local distances, are more conserved. So this is a very different underlying idea compared to PCA. Still, the output of these approaches, and one of them is what I show here, TSNE. The output, in a way, you can compare with what you get with PCA. Only the difference is that PCA is a linear technique, and then these newer approaches like TSNE or UMAP is another example. They are not linear. But here you see one example, and this is an example of a set of different images, and each color indicates a different type of image. And you can see here that PCA really cannot make a good representation of this data in two dimensions, whereas this TSNE approach very nicely shows different groups. Each color here is really separate from all other colors. And this is an example which is more relevant from, indeed, this single-cell sequencing data, where we have, this is just one randomly chosen example of an Arabidopsis sample, where we have over 4,000 different cells that were analyzed. And then you can see that the application of TSNE really, again, separates nicely related cell types. So it's a technique that can really help you to get insight into your data. Now, one other reason why this type of unsupervised analysis is also very relevant relates to what we call embeddings. And embeddings are ways of representing your input data. So you can think kind of like making features of your data. But these embeddings, they are made by analyzing large sets of available sequences, for example, or structures. And I show here some examples to papers where people present some examples of embeddings, one for at the DNA level and one at the protein sequence level. And the thing here is that by using lots of available sequence data to make these embeddings, so this is again done in an unsupervised way, but by making use of that large amount of data, you can get a representation of your sequences that you can subsequently use in supervised machine learning. And often you will have a better representation than if you start from scratch. So it's really in many applications has been shown that using this type of unsupervised based generated embeddings can really improve your prediction performance. Now, I think that I've only a few more things that I want to share with you. And one thing here is that I want to get back to this picture that I showed at the beginning, which was about different scales of data that we deal with. And I focused a bit on the genomic scale, but of course, there's much more than that. And you can really think about connecting genomes to traits. So I want to briefly talk about that. Particularly, you can think of doing what is called genomic prediction, where you have a population for which you have genotypes and phenotypes. You use that to make a prediction model, and then for a population of interest, you can try and predict phenotypic values based on genotypes. Now, this is a very classical statistical problem, so there are many approaches that work quite well, but it's also under active development how we can use machine learning towards this end. And in particular, people are focusing a lot on using deep learning. I think if you have time, I will not discuss this example here in detail, but I want to mention that there are different ways in which people represent genome data as input for deep learning to do genomic prediction. And this is a recent model that I think had quite promising performance. So if you're interested in this type of applications, then you could look at that in more detail. We're also planning to go in that direction, or we started to go in that direction ourselves. What we so far have mainly been doing is to analyze a bit under which conditions more classical statistical approaches work well versus under which conditions machine learning based approaches for genomic prediction work well. So you see here some visualization of how performance on a test data, this is based on simulated data, how that depends on if you have a simple trait or complex traits, and then also how many samples you have. And what this basically shows, and this is also what you would expect, is that there's a clear effect, of course, of the number of samples. So if you go from left to right here, you see that with fewer samples, of course, your problem is more difficult. And also the trait type has an influence. But what this also showed quite interestingly is that there are some scenarios in which the more classical approaches really underperform compared to machine learning approach like a random forest in blue, or a boosting approach, which is kind of related to random forest. So this kind of an initial investigation, but we really think it's an interesting area to continue with. One issue is that compared to some other applications of, in particular, deep learning, the number of training cases that is available is often not so large, at least not data that's available in the public domain. So this is something that we're still struggling with a little bit.
Dr. Alti, do you have 5 minutes to finish your lecture? Is okay?
Yeah, this is very much okay, because actually I think this is my last slide, so maybe I will not even
great.
need. I will maybe not even need 5 minutes, but I will take my time to talk about this.
Okay, go ahead.
Yeah, Thank you. Yeah, because this is actually one but the last slide. The last slide is with the references, so I will not. I don't need to show that now. So I want to briefly summarize what I talked about, and I want to say something about what I think are important directions for the future. So first, what I've been talking with you about is how we can apply machine learning to analyze plant omics data, and we have this application more at the level of genome regions, or we have the application more at the level of genes. And then I also talked about some recent developments, the use of unsupervised analysis and using protein structures as input, for example. One key thing that I want to again mention is that the interpretation of models is often quite important in our field. So this is not always the case in machine learning. In some other applications, when you have a self-driving car, for example, you really don't care why your model makes certain prediction, as long as it is correctly identifying people on the street and these kind of things. But for application on omics data, often you want to understand more about why the model is making certain predictions. That being said, let me briefly talk about some future outlook. I think that one thing that you can see happening in, let's say, data science in general, but also in the plant sciences and data sets that are generated there, is that there's increasing awareness of making data better available and using standards to describe not only the data, but also the metadata. And I think this is something that will really also be beneficial for the application of machine learning, because there's so much data measured, but lots of what is published is not really usable. For example, if the experimental conditions are not well described. And I do see, although it's maybe going slowly, but I do see that there's a direction in the right movement where standards are increasingly seen as important. One other thing that I think will be very interesting to work on is the application of machine learning to less controlled environments, let's say on data from the field, for example. And this is something that I've not been involved with, but I've seen some other examples, and I think it's really, of course, this is key also towards application of machine learning in the context of breeding in the end. A third thing that I didn't have really time to talk about, but is very interesting, and also there's lots of development there, is that people also have more and more attention to what you can call unstructured data. So the examples that I gave of machine learning today are really structured in the sense that you have a set of cases and a set of features that you calculate. But there are also other examples and where the input data is less well structured. And one example here is that I give of something that we're looking at ourselves now is if you have some pictures about the insect that moves on plant leaves. So you have this insect moving on different genotypes, and you have images measured for all these different genotypes of how the insect moves over this plant leaf. Then how do you use that kind of data as input, for example, for genomic prediction? And it's quite of an interesting direction to go towards. And one final thing I want to mention that I think is also related to model interpretation is the use of machine learning models to also make decisions. There are different types of machine learning that allow you to do that. Reinforcement learning is one of them, and this is not so often used yet in plant omics data analysis, but I do think this is also something that you can expect more in the future. So that being said, I think I want to finalize here. I want to thank you for your attention. It was really a pleasure to give this lecture. So thank you.
Thank you very much, Dr. Alti, for the excellent lecture. It is a lot of information of the great importance for the area. The use of machine learning in sequence, population genetics, active genes, among other that you mentioned, are great advances for the science. So, for the participants, unfortunately, Dr. Alti will not be able to participate in the round table. As you saw that he has a flu. So if you have any question or curious about his talk, you can do this now. Let's see some question. We have some question for Dr. Alt. the audience of the YouTube channel. We have from Jacqueline Schmidt, great lecture, Dr. Alt. It is possible to include together data from different experiments, biochemical, genetic, morphological, to predict responses of a pathway acting in a given species.
Yeah, thanks for this question. I think it's an excellent point. So I didn't talk so much actually about this kind of data integration, but it's for sure something that people work on. One problem often will be that maybe your training data will be quite heterogeneous, so it might not be so easy to combine data because maybe the conditions were different from one experiment to the other. So you really need to think of ways to not just have the measurements themselves, but also somehow represent some features of the experimental conditions. Let's say, so you can think of including some. Let's say if temperatures were different or something. I'm just giving an example here, but you can think of including that type of information also as features in your machine learning model. So indeed, I think this is possible, but it's not so. It's not so easy. Also because of the heterogeneity of the data, you maybe need quite some training examples. But it's something that makes a lot of sense. Yeah.
Okay, thank you. Do you have more question? So I think no. It is dark in your room. It is night here in your city. Yeah.
I had not realized this, but indeed already the evening is starting here, and yeah, that's true.
Okay. So thank you so much, Dr. Alt, more time for accepting our invitation, and we wish you well. It's okay. And for the audience, if we have some doubt or some question, we can pass your contact for the people contact you.
Yeah, I would very much appreciate that. I'm very sorry that I cannot join the roundtable, but I think I better not do this. But I'm very interested. If you have questions, really feel free to contact me. No problem at all.
Okay.
Yeah.
Thank you
Thanks a
so
lot.
much.
Yeah. Okay.
Okay.
Bye then.
Bye. So, continue the event for the second lecture. I would like to invite Dr. Ricardo. Dr. Ricardo is an assistant professor at the Department of Computer Science at the Federal University of São Carlos, leading the Bioinformatics and the Machine Learning Research Group, supervising both bachelor and graduate students. He is also a researcher on productivity at CNPq Level 2. Thank you very much for accepting. Can you hear me?
Hi.
Can you share your screen?
Yes, just 1 minute.
Okay. Okay, I can see. You can start. The audience is yours.
Okay. Thank you, Rosimara. Thank you, everybody. It's a pleasure to be with you here. In this talk, because of the topic of the conference is computational intelligence, with which algorithms have contributed to improvements in agriculture use, okay? So I'm going to present you here a random forest algorithm. And I'm going to tell you how you can use the random forests, okay, for SNP selection in rice. Because I think it's very much easier to introduce an algorithm with an illustrative example. I will not use a biological example for introduce you the algorithm, okay? I'm going to work with a very toy problem, okay? A movie data set. but you can extrapolate this kind of data for all data you would like to extrapolate it. Okay, so we are going to work with biological data, but here to introduce the algorithm, I think it's much better to use this toy problem. Okay, so we have a data set here where each sample is a movie, okay, and each feature is a country, star, or gender, and we want to predict if the movie is going to make success or not. Okay, if we think about a biological data, okay, each feature here can be, for example, I don't know, a SNP, okay? And the target attribute can be, for example, the productivity of rice, okay? So, a random forest is made of decision trees, okay? So what's a decision tree, okay? This is a decision tree. So in decision trees, internal nodes represent features, okay? Edges represent feature values, and leaf nodes represent target features. Okay, so we want to build an algorithm to construct this kind of tree so we can make decisions on our data. So when we talking about selecting SNPs, okay, so each internal node here will be a SNP, okay, an SNP. And then the tree will contain the most important SNPs for a given problem. For example, predict the productivity of the rice. Okay, so a decision tree is used to classify instances. So the objective is to classify the larger number of instances with the smallest possible tree. If we have the smallest possible tree, it is supposed that this tree will contain the most important features of my data set. In our case, the most important SNPs, okay? So it's the induction algorithm to build a decision tree is a recursive algorithm. So the induction algorithm recursively partition the training set until the subsets obtained with the partitioning contain data from a single class or data from a pure type of prediction. So for example, if you want to predict the productivity of the rice, we want to produce subsets of samples of rice that have the same productivity, okay? Okay, so going back to our toy example here to see how a decision tree works. Okay. So if we select the feature country to start constructing our decision tree, okay? What do? We create a node country with the feature country, and then we create edges for each values of that feature. So if we go back here to our toy data set, we see that for the feature country, we have 3 possible values for this feature country, okay? USA, Europe, and other. So if we choose the feature country, how is it going to divide our data? So we are going to check this. Okay? Looking at the classes or looking at the value of the target attribute we want to predict. Okay, so country, if country is equal to USA, we have 3 movies that are going to make success and 1 that's not going to make success. The same for Europe, the same for other. So if we look here at these subsets, we can start making some conclusions. Okay, so if we look at the star feature, okay, if we use the star feature to divide our data set, okay, how does the star feature divides our data? Okay, so if star is equal to yes, we have this kind of division. If a star is equal to no, we have this kind of division. Okay, what if we took the feature Geiger? Okay, so if we get the feature Geiger, it has 3 possible values, and it divides our data set according to these 3 subsets. Always remembering here that we have movies that are going to make success and movies that are not going to make success, okay, depending on the value of the feature Geiger. So what feature to choose? We have to look at the subsets that are created with the division of the feature. Okay, so if we look at, for example, here in the subsets created by the feature country, let's try to see how are the subsets that were created. Okay, if we look at this subset here, okay, so for the value, sorry, if we look at this subset here, for the value other for the feature country, we have that we have only data for the class no. So this is a very good division. Okay, if we get the value USA, okay, the value USA results in a data set with 3 samples from class yes and 1 sample from class no. So we have much more samples from class yes than from class no. Okay, it's a good division. However, if we, for the value Europe, the subset of instance generated is not that good, okay? Because we have 2 samples for each class, so we cannot conclude anything. So the objective of building a decision tree is choosing features to be put in the tree so that these features generate subsamples that are pure, okay? In this case here, subsamples where movies belong to only one class. In case of productivity of rice, subsamples where the samples there in that subset belong to have the same productivity, okay? Belonging to the same class of productivity, for example. They are all very productive. So looking at the tree, we are going to know what features, in the case of the rice, what SNPs are going to be good for a high productivity of the rice, okay? So we are going to make this analysis for all features of our data set. Okay, but how to choose, okay, a good feature to be put in a data set, to be put in a decision tree? Okay, we have to use statistics. So statistics has measures that allows us to evaluate these previous situations here, to see which feature is the best one to be put in the decision tree. Okay, so we are going to use entropy, okay, which is a measure that measures the uncertainty of a random variable, okay, and quantifies to what extent the data are spread out over its possible values. Okay, and we are going to use information gain. The information gain calculates the reduction in entropy. So we are going to select the feature that offers the higher information gain. Okay, so we are going to calculate the entropy for a data set, and then we are going to calculate the information gain for each feature and select the one that reduces the entropy the most. Okay, so the entropy, the higher the entropy, okay, the more confusion I have in my data. So we want to select a feature that decreases entropy. So the feature that decreases the entropy gives me the highest information gain. So that's going to be a good feature. Okay, how do we calculate entropy, okay, for a data set? So if we have a data set with 2 classes, positive and negative, okay, we calculate the entropy by this formula, where P1 is the probability of observing the positive class, and P0 is the probability of observing the negative class. Okay, so entropy value is closer to 0, low entropy, means that the data is concentrated near one value. So think about the productivity of rice. We want to find the best decision tree, okay, to predict productivity of rice. So a good decision tree will have subsets of samples in my data set where, subsets of samples in the decision, in the leaf nodes of the tree. So these subsets of samples, okay, in the leaf nodes of the decision tree will be concentrated nearly one value of productivity, okay? High productivity, low productivity, so then we can distinguish between different samples with different productivities. Entropy is close to 1. High entropy means that the data is spread out as much as possible, so there is confusion in data. I cannot make any decision. So we want to find a feature that decreases entropy. Okay, so the entropy for an entire data set okay, is calculated this way. So getting our toy data set, remember we had 5 movies, 5 from the positive class and other 5 from the negative class. So we calculate the probability of the positive class, the probability of the negative class. Okay, when we put this in the formula, we have entropy equals to 1, which is the highest possible entry for this data set. Okay, there's total confusion in the data set. 5 instances from one class, 5 instances from the other class. So now we want to find the features that decrease this confusion. Okay, so the information gain of a feature now is going to reduce the entropy, okay, when I select a given feature. So I want to choose the feature with the highest information gain. So in our example, the information gain for each feature is going to be given by these formulas here. So the information gain for country is going to be the entropy for the entire data set minus what I'm calling here the remaining entropy for the country feature. The remaining entropy for a given feature is the expected remaining entropy after testing that feature. So we're going to select the feature that gives the higher information gain. So think about that. When I get my original data set, the entropy is very high. Okay, so now suppose I choose a given feature A. Okay, and now I'm going to calculate entropy for that feature A. Now suppose the entropy of the data set after selecting the feature A is very low. Okay, so I'm going to subtract the original entropy from this remaining entropy, the entropy of the feature A. So I'm going to subtract a very small value from a big value. So the result is going to be information gain very high information gain. That's going to be the best feature to be chosen. Good. So if we have a feature A now with D distinct values, okay, this feature is going to divide our training data into E subsets. Okay, each subset has, of course, positive and negative instances. So the remaining entropy for a given subset, okay, is going to be given by this formula, where we are going to get the positive instances in a subset divided by the positive and negative instances in the subset, okay, for the calculation of the positive and the negative, okay? So this is going to be the remaining for a given subset, and we are going to have to calculate this for all k subsets. So to calculate the expected remaining entropy for all values for a given feature, okay? So I'm just checking if feature A is a good feature to separate my data. We have here to calculate the expected remaining entropy when we select feature A. So this expected remaining entropy is calculated by this formulation, okay, where we are going to calculate here the entropies for all subsets of features, all subsets of samples generated when I choose, okay, generated by one division that's performed with feature A, okay? And then the information gain after selecting feature A is going to be the information gain for the whole data sets minus the information gain for the feature. And we are going to do an example with this. Okay, let's perform a calculation with an example. So let's get the entropy for the feature country, okay, and let's calculate it. And then we're going to perform the same thing for the other features to see which feature is the best one to construct our decision tree. So if I get the feature country and see how this feature divides my data, what I have to do now is to calculate the remaining entropy here, okay, the entropy for each subset. So the entropy for the subset that resulted from using the feature country to divide the data, and the subset resulted from the feature country being equal to USA, the calculation is going to be like this. Okay, so the entropy for the feature country being equal USA is 3 divided by 4, log 2, 3 divided by 4, okay? And 1 divided by 4, log 2, 1 divided by 4, okay? So minus this, okay? The resulting calculation is going to be this value, okay? And we are going to perform this calculation for all the subsets. So this subset now, so I'm doing the calculation for the positive and negative instances, putting in the formula here and getting the entropy. So look, the entropy for this subset is the highest possible entropy for it. So it's 1. Okay. So when I look at this subset, I can make no conclusions about the data. When I look at this subset, I can make some conclusions. Okay, most of the data belong to the positive class. So probably if an instance is similar to the instances here in this subset, so it's going to belong to the positive class. Okay, now I calculated the entropy here. The entropy here is the smallest possible entropy, okay, because this subset is pure. Okay, okay, good. Now, how do I calculate the remaining entropy for the feature country? I have just to sum up all the entropies calculated for each subset constructed using the feature entropy. Okay, so we have to do 4 divided by 10 feature, 4 divided by 10 multiplied by the entropy USA. Why 4 divided by 10? Because if we look at here, we have 4 examples, 3 plus 1 divided by 10 because all the data set has 10 samples. Okay, so it's 4 divided by 10, the entropy in this subset, plus 4 divided by 10, 4 divided by 10, so multiplied by this entropy, plus 2 divided by 10 multiplied by the entropy calculated for this subset. So the remaining entropy for the feature country is 0.7244. So we are going to perform these calculations for all the other features. Okay, so we got the feature country, we check the subsets created when using the feature country to divide the data, we calculated the entropy in each of these subsets, we sum up these entropies, and the result is the remaining entropy for the feature country. So we're going to do the same for the other features. Okay, good. But first, having the remaining entropy, of course, okay, for the feature country, we are going to calculate the information gain for the feature country. So the information gain for the feature country is 1, which is the information gain for the whole data set, minus the remaining entropy for the feature country, which gives us the value 0.756. Okay, so keep this value in mind because this is the information gain of feature count of the feature country. Now we are going to perform these same calculations for the other features. Okay, so getting the feature star, remember feature star has 2 values. So the entropy when choosing value yes is 0.9852, and the entropy when choosing feature star with for the value no is 1. So the remaining entropy, okay, is 7 divided by 10 entropy yes plus 3 divided by 10 entropy no. Why 7 by 10? Because I have 7 samples here, and 3 by 10 because I have 3 samples here. Okay, from my data set, 3 samples have the value no for the feature star. Okay. Now the information gain for the feature star is 1, which is the information gain of the whole data set minus the entropy, the remaining entropy for the feature star. So the information gain when choosing the feature star is 0.01. Now we're going to do the same calculation for the feature gainer. Okay, the feature gainer has 3 values, so it creates to me 3 subsets. So I calculate the entropy in this subset, this one and this one. Then I calculate the remaining entropy with the summation of all these entropies, okay, multiplied by the respective values of the probabilities of observing the samples here in the subsets. So 6 by 10, 1 by 10, and 3 by 10. Okay. So I have a remaining entropy of 0.8264664. And the information gain for the feature gainer is 1, which is the information gain of the whole data set, minus the information gain of the feature gainer, which is equal to 0.17. So we have the information gain of the 3 features of our data set. So which feature to choose? The feature that maximizes the information gain. So the feature that has the maximum information gain is the feature country. Okay? So we are going to choose the feature country to start dividing our data, which means to start constructing our decision tree. Okay. Now we are going to perform the same calculations again, okay, with all the features, and I'm going to simplify here, because if I already chose the feature country to be put in my decision tree, now I have to analyze the other 2 features, gender and star, okay? So I have to choose which features to put here in these subsets, okay? Now this subset is my new entire data set, okay? Now I'll have to calculate the entropy for this data set, okay? Considering it now as an entire data set, and calculate the remaining entropy for choosing feature star to divide this data set, and for choosing feature gainer to divide this data set. Calculate the remaining entropies, and then calculate the information gain for choosing each feature. Okay, again. So I'm going to do these calculations for this subset, and this subset I don't have to do anything because it's already pure. Okay? So that is why the algorithm is recursive. It calls itself on new and smaller subsets of data. So I'm dividing data recursively. Okay, so because now we know how to perform the calculations, we're not going to perform the calculations all again anymore. Okay, what I'm going to do is using the intuition with you. Okay, so if I get this data set here, okay, let's see what features are going to be the best one to divide this data here, okay? If I select feature star to put here, it will generate these 2 subsets. And if I select feature gainer to put here, it will generate these 3 subsets, okay? And 1 is empty. So which is the best division? Of course, this one, okay? Because choosing this feature to put here, I'm going to construct pure subsets. So the information gain for feature star is going to be higher than the information gain for feature gainer, okay, considering this subset. So star is going to be put here, okay? And this is my new data set. This is my new decision tree, sorry. Now I have to do the same calculations with this subset here. So which feature I'm going to choose to put here? Star or gainer? If I choose to divide this data set here using feature star, the resulting subsets are going to be like this. If I choose feature gainer to be put here, okay, the resulting subsets are going to be like this. So which feature is going to give me the higher information gain? This one, gainer, because it produces the more pure subsets, okay? It produces only pure subsets. So this feature is going to be put here. And this is the final decision tree, okay? So with this toy data set, I can show you how to use a decision tree, okay? How to use an algorithm to construct decision trees, okay? To a given prediction target, okay? A given prediction task. So we can extrapolate this for SNPs, okay? So each feature here, country, star, or gender, could be a SNP, okay, with its all different values, okay? But the process would be the same, okay? So suppose I want to construct a decision tree, don't know, to predict the productivity of rice. I want to predict different classes of productivity. high productivity, medium productivity, small productivity. Okay, or I want to product, for example, the strength of my grain. Okay, very strength, not so strength. Okay, and different targets you can imagine for the rice. Okay, so the calculations are going to be the same. So I think it's easier to show this toy problem, and then you extrapolate to any kind of features and biological features you want. Good. Now, having this decision tree already induced, okay, the final decision tree, we can use it to predict the values for new movies in this case, for new samples of any kind, okay? So, getting, for example, here, a new movie, okay? The values of its feature, of their features are, for example, for movie 11, country USA, star no, and gender fiction. So what's going to be the class of this movie? Okay, if the country is United States and the star is no, so the movie is not going to make success. It's false. And we are going to perform the same analysis for the other movies. Okay. And here, I have a new prediction. Good. Okay. By now, I showed you how to construct a decision tree when my target attributes are categorical. Like I told you, classes of productivity, classes of a movie, okay? But what if the output I want to predict is numerical? So for example, the value of the productivity of the rice, okay? Now, the leaf nodes, okay, will contain a mathematical function and not a target and not a class anymore, okay? In the simplest case, okay, the prediction the prediction of the decision tree, okay, can be computed for each leaf node as the average of the values that fall in that leaf node. Okay, so for example, if I want to build a decision tree to predict the productivity of rice, okay, so the prediction for each leaf node, okay, is going to be the average of the production targets of the instances that fell in that leaf node, okay? I'm going to compute this average for all training instances that I used to induce my decision tree, okay? So I'm going to get the average of all these training instances for each node, okay? And these averages are going to be the predictions for the decision tree. Or I can build a linear model, okay? I can fit a linear model in each leaf node. I can fit a linear model using the examples of each leaf node, okay? So this mathematical function is going to predict the value of a target attribute. So for example, here, I have another toy example. So I have, I don't know, it can be here, vehicles, no? Suppose that each row here is a vehicle, and as features we have the potency, the weight of the vehicle, the acceleration, and we want to predict the consumption, okay? So again, each feature here could be a SNP, each row could be a sample of rice, okay? And the target variable could be the production of that sample. So in this case, we fit a regression tree, okay? Where here in each leaf node, okay, instead of calculating the majority class of an instance in each leaf node, we calculate the average of the target attributes of the instances in each leaf node, okay? The average, or we fit, or we get the instances that fall in each leaf node and calculate here and fit a linear regression, okay? So fitting a linear model, for example, we could have in each leaf node a linear model like this one, for example, that predicts the consumption based on this linear model. That's a value multiplied by the potency plus a value minus a value multiplied by the wage plus here a constant, okay? So this is a linear model, okay? Beta 0 plus beta 1 times a feature plus beta 2 times a feature plus beta n times the enhancement feature. Okay, so we're going to have one of these for each one of our leaf nodes. Okay, now that we know what a decision tree is, okay, we can give the definition of a random forest. So a random forest is a collection of several decision trees. Each decision tree is going to be trained on a subset of instances from the original set, what we call a bootstrap sample. So each decision tree here, okay, is going to have an output, as we saw, because we already saw how to train an individual decision tree. So we know that each decision tree in the forest is going to be its own output. So multiple outputs are going to be used to decide the final prediction for a random forest. Okay, so suppose I have a random forest, I don't know, with 1,000 trees. Okay, and I want to predict the productivity of rice again, if I have a high productivity or a small productivity. Okay, so given a sample of rice, when I give this sample of rice to a random forest, the output is going to be computed based on the outputs of all the 1,000 decision trees. So we can use majority voting. If the majority of the decision trees tells that that sample has a high productivity, so the prediction is going to be a high productivity. Okay, and we are going to see how we can use the decision trees to actually select the best features of our data set. So in our case, the best SNPs related to the productivity of rice. Okay. So the idea to build a random forest, okay, is that a large number of uncorrelated trees working as a committee get better results than any single tree. So here, having our original data set, for each decision tree that we are going to induce, we get a bootstrap data set. A bootstrap data set is a data set constructed randomly picking, okay, some samples from the original data set. Okay, and also, okay, these data sets we're going to see, they can be constructed using subsets of the features as well. Okay, so I randomly get some instances and also randomly get some features to construct the bootstrap data set. And then we induce our decision tree. For each decision tree of our forest, we're going to have a bootstrap data set. Okay, and then we induce the decision tree in the bootstrap data set. Okay. So the features and the instances that were not selected to form, okay, not the features, of course, but the instances. The instances that were not selected to form the bootstrap data set are called the out-of-bag data set. So for each decision tree in our random forest, we are going to have a bootstrap data set and an out-of-bag data set. So after training the decision tree in the bootstrap data set, I can evaluate this decision tree in the out-of-bag data set and calculate, for example, the mean square error of this decision tree, the accuracy of this decision tree, okay, depending on my task, if it's classification or regression. Okay, anyway, so as I told you, each tree can be constructed from a random subset of features or attributes here, okay? So the idea is that in a random forest, we have many different trees from each other, and that the combination of these trees make a strong model. And then, for the classification, the final prediction can be obtained using majority voting, as I told you, and for regression, the final prediction can be obtained averaging the individual predictions, okay, averaging the individual predictions of the trees in my random forest. So example here, as an example, I'm getting a new sample, okay, where the features are age, income, sex, and merit, and I want to predict the I think I want to predict remember the target attribute. I think it's married. I want to predict if it's the person is married or not. Okay, so for example, so most of the people, okay, or most of the data sets, most of the trees in the forest predicted that the person is not married. So the final prediction is that the person is not married. Okay. Okay, good. So remembering the out-of-bag sample that I told you, so we can use its error to select features. So that's a very good characteristic of the random forest. So for each tree, the error of the out-of-bag sample is going to be calculated. Okay, error rate for classification, mean squared error for regression anyway. And the same is going to be performed after permuting each feature. And then the difference between the two is going to be calculated and averaged over all trees. Okay, so for each tree, we are going to get a feature in the out-of-bag sample, and we are going to change the values of that feature in the out-of-bag sample. Okay, so we are going to calculate, for example, the accuracy of the tree for classification problems, the accuracy of the tree in the out-of-bag sample, and then we are going to get a feature. Okay, let's evaluate this tree. Let's evaluate how good is a feature A for this decision tree. So we go there in the out-of-bag sample, and we shuffle all the feature values in the out-of-bag the sample, okay, for the feature A that we're going to evaluate. And then again, we evaluate the tree in that data set. Okay, if the accuracy of the decision tree decreases when I shuffle the data in the out-of-bag sample, that means that that feature was important, because the difference between the accuracy of the tree in the original out-of-bag sample and the accuracy of the tree in the shuffled out-of-bag sample is high. So that feature is important, okay? Because it decreases the accuracy, it decreases the performance of the tree after I shuffled it. So I'm going to do this for all the decision trees that have that feature, and I'm going to average, okay, this difference in accuracy over all features. So this is going to be a measure of the importance of the feature for my data set. And then I can rank all the features based on this measure, okay? And then I can verify what are the most important features in my data set for that given task. For example, the productivity of the rice. Okay, here are some useful links I would like to leave you with. And I'm going to show here to you what I would like to show is how to apply this. Okay. I'm not sure it's, let me increase here. Yes. I hope everybody's seeing. Okay. So here's a SNP data set. Okay. So I'm using R here. Okay. And I just loaded this data set here. Okay. And I'm showing you the SNPs. Okay. The values of the SNPs. And here is the target variable. So in this data set, for example, given here, this is part of a data set. Okay. This is a huge data set, but I just extracted a little part of it. So I have 50 SNPs. Okay, and I want to predict the productivity of the rice. Okay, so as you can see, there are some values missing here. So what I did here is that I constructed some functions that just replaced the missing values. Okay, so for example here, missing values with the SNP value here, the nucleotide that appeared the most in each column. Okay. Just to use this data set with machine learning. Okay, now. Okay, very good. So I'm showing here just the mode, okay, the SNPs that appear the most, okay, in each column where I have SNPs missing. Okay, so for example, SNP 5 here. SNP 5, yeah, SNP 5, there's a SNP missing. And the SNP that appears the most in 5 is in the column responding to SNP 5 is T. So I'm going to replace this guy here by T. Okay, just to use in the decision tree. Okay, good. So what I'm doing here, I'm dividing my data set, okay, and training and test data sets. And with this package here, I can construct a decision tree for my data set. So I'm building a formula here. What I'm doing, I'm saying here that I'm going to use all the features to build my decision tree against the target feature, okay? With this command here, this function, I can construct a decision tree with the training data set. Remember, I divided my data set here into training and test data sets. And here is the resulting decision tree. Okay, so it's a decision tree for predicting the production of rice. Okay, so depending on the value of each SNP, okay, I have a productivity being predicted. Okay, so I can consider that for just one decision tree, these are the most important features of those 50 features in my data set. And using this decision tree, I can make predictions using the test data set with this function predict. Okay, I don't know if everybody's familiar with the R function, with the R program language here. And what I'm doing here, I'm constructing a table where I am combining the true productive values with the predicted productive values. And as you can see here, I have the predictions, okay, using a very simple decision tree for each sample of my test data set. Okay. Okay, and here you can see I just calculated the squared error. Okay, the root square error for this data set. Now I'm going to use the other package that's called rpart to construct another decision tree. So the procedure is going to be the same. Okay, I'm going to use all targets to construct a decision tree for the target data set. Okay, and here, oh, sorry, yeah, the rpart function was called here, and here is my decision tree. So I'm going to plot, okay? I'm just showing the decision tree here, but instead of showing it this way, I'm going to plot the decision tree here with this library here and this function. So this is the decision tree I just constructed with the other package, rpart. So because this is a more sophisticated package, okay, and its hyperparameters are different, I have a bigger decision tree here. Okay, so the bigger decision tree, the bigger the decision tree I have, more complex it is more difficult to interpret. So what if I want to decrease this decision tree? So what I can do actually is to verify the errors obtained every time I increase the size of the tree. Okay, so let's get the smallest error here. So for example, here in the level 4, okay, I have an error that is the smallest one among all the errors here. Okay, so what I'm going to do is that I'm going to prune my decision tree based on this error. This error was calculated while inducing my decision tree in the training data set. Okay, so I'm going to actually, yes, and I got here the level 6, okay, but I could have taken the level 5 here. Anyway, what I'm going to do here is that I'm getting here, look, I'm getting the table, okay, in the column CP, see, CP, that's complexity parameter, and I'm getting the complexity parameter in line 6, okay, line 6 is here. So I'm going to prune my decision tree, okay? After this, the split number 6. So applying the pruning here, I have a smaller tree. So compare this tree with this one, okay? And the prediction, okay, the evaluation measure comparing the pruned tree with the original tree are not so different. So now I have a less complex decision tree from where I can get the most important snips from my data set, okay, and together with a higher interpretability. Okay, I can convert this to rules more easily. And here I'm just comparing the predictions of the original decision tree with the pruned decision tree, and calculated here the evaluation. See, the pruned decision tree had a smaller error than the original decision tree. Okay, so these are decision trees, but how can we select very good features using random forests? Okay, so again, I'm loading here my data set. Okay, the same data set I used for decision trees. Okay, I'm making a function here. Okay, I'm making a function here again to fill missing values with the SNPs that appear the most in the data set. Okay, nice. So here I have data set with no missing values. Now I'm going to use a library called random forest. Okay, and a package here to make some plots. So what I'm going to do, I'm going to perform 100 executions of random forests. Okay, so I will have 100 random forests. Okay, and I'm going to build a rank for each random forest. For each random forest, I'm going to have a rank with 10 positions. In this rank, I'm going to put the most important features for a random forest. Okay. And here I'm building a matrix, okay, that's giving to me the following. For example, see, any row, number of execution. So this is a matrix with 100 rows per 10 columns. So for each execution, okay, so we're going to have 100 random forests. So for each random forest, we are going to have a ranking with 10 most important features. Okay, good. So this is a training loop where what I do is that I execute a random forest. Okay, I separate my training data here in training and testing. And then I execute a random forest, see, with 50 trees, okay, calculating the importance of each feature. And the importance is calculated based on that mean decrease on the mean square error. Again, so I have a tree in my data set. Okay, I calculate the mean square error of this tree in the out-of-bag sample. Okay, and then I shuffle the values for one feature and calculate the mean square error again. If the mean square error increases a lot for the shuffled data set, then that's an indicative that that feature is important. So I'm going to perform this kind of calculations for all 10 executions or all 100 executions for 100 decision trees. And I'm going to store here, okay, the more important features, the 10 more important features according to this importance calculate that I just explained for each execution. Okay, and at the end, what I have here, see, I'm just showing the head of that data, of that matrix. I just showed you 100 per 10, okay? So for each execution, for each random forest, what I have is a ranking of the 10 most important features for that data set and for that random forest execution, okay? And now, what I did here, these are the errors for each of the executions. And what I did now is that I verified all the SNPs that appeared here. Okay, because I have repeated SNPs here, of course. See, for the first execution, SNP 38 was the most important one. For the second execution, SNP 38 was again the most important one, and so on. Okay, so if I get this matrix and use the unique function, I can obtain all the SNPs that are here. So these are the most important SNPs for my data set. Okay, very good. And what I'm doing here is that I'm going to plot a graph, okay, with this ranking. Okay, this plot, okay, what I'm doing here is that I am counting how many times each feature was considered the most important in each ranking. So from 100 execution, SNP 38 was considered the most important, okay, first position in the rank 0, 86 times. SNP 26 was the second most important in the remaining 14 times. The other SNPs did not appear in the first position of the ranking. Okay, and I am calculating this, see, for all the ranks, all the positions of the rank. So here, for example, for the rank 1, for the second position of my ranking, SNP 38 appeared 14 times in the first position. SNP 26 appeared 84 times, okay, in the second position, okay. SNP 38 appeared 14 times in the second position of the ranking. SNP 26 appeared 84 times in the second position of the ranking, and so on. Okay, so I have these calculations from R0 to R9, how many times each instance appeared in that ranking, that position of that ranking. Okay, good. And here I can plot a graph, see, showing the most important features. So for example, SNP 38, as we saw here, okay, it was the most important feature. See, SNP 38 appeared 86 times from my 100 executions of random forests. SNP 38 appeared 86 times as the most important feature. Okay, so in the first position of the ranking, SNP 38 appeared here 84 times. That's why the color blue here, the bar blue, is high. The other 14 times, okay, were occupied by SNP 26. SNP 26, this color here. See? And the same analysis was performed by the other positions of the ranking. So as the positions of the ranking, okay, are becoming, as I get far from the first position of the ranking, more SNPs share that position of the ranking. Okay, so this is a visualization, okay, graph to see what are the most important SNPs for the productivity of rice, okay, using random forests. To validate more statistically my experiment, of course, I have to increase here the number of times, the number of executions. So instead of 100 executions, I would perform 1,000 executions, 10,000 executions. Okay, so then I validate more statistically my experiment. Okay, and having the random forest, of course, I can make predictions for new samples of rice. Okay, so I'm getting here my random forest model, see, and I am doing. I'm getting a random forest. Okay, of course, I made 100 predictions of random forests. Okay, but in order to make a prediction for a new sample, I'm going to execute one random forest again. Okay, because otherwise I would have a committee of random forests, but I want to make the predictions using just one random forest. So I induce a random forest model again using the training data set, and here I am predicting the value of the productivity for that using that model. okay, and using the test data set. And what I'm doing here is constructing a table comparing the predictions and the true values, okay, here. The true values of the production and the predictions for the productivity of the rice. And if I compare here the error of the random forest compared with the error of the decision tree, see, the error of the random forest is smaller, okay? And what I can do, of course, is that I can save, okay, all this information and then use this model then again for here for making predictions. Okay, yeah, that's it. I think it's more or less what I wanted to show you. So now I hope you understood how you can build a decision tree. Okay, and I hope you know the mathematical foundation behind building a decision tree. Okay, that can be used to predict targets for any kind of biological data and can be used for the rice, which was our case here. Knowing how to build a decision tree, you now know how to build a random forest, okay? That's just building hundreds of decision trees using out-of-batch samples. And now, use for each decision tree of the forest, you can compute its error based on the out-of-batch sample, okay? And then you do this for all the features in the decision tree, and you average, okay, this out-of-batch error, the difference between the out-of-batch error and the out-of-batch error when you shuffle the feature, you average these differences over all the decision trees in the forest. And then you rank the features based on this calculation of the importance of each feature. And having this ranking, okay, you can indicate the most important SNPs, for example, for, in our case here, the productivity of the rice, okay? This is just to finalize, this is a project, okay, that we are doing together with Embrapa, Arroz e Feijão. They are providing us the samples of rice, the target values they want us to predict, and then we provide them with the most important features we find, okay? And then they have to validate this in lab, okay? I think that's it from my side. If we have questions, I'm very happy to answer.
Thank you, Dr. Ricardo, for the excellent lecture, and you show for us many information about the decision tree, machine learning, and the handle forest. And the question will be in the round table of the end of the 30 lectures, okay? So now we'll break, it's okay, about 15 minutes. So guys, take this time to drink your water and a coffee. We're back out at it.
bash, it's a gas, gas, gas. Get down. I was raised by a toothless bearded hag. I was schooled with a strap right across my back. Yeah, but it's alright now. But just a gas, it's alright. Yeah, jumping jack flash, just a gas, gas, gas. I was drowned, I was washed up and left for dead. Yeah, I was drowned, my feet, my soul, they bled. Yeah, 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 I was drowned at the crumbs of crust of bread. My, I was drowned with a strike right through my head. My, yeah, but it's alright now. That's just a gas, 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 but it's alright. Jumping jack flash, it's a gas, gas, gas. Jumping jack flash, it's a 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 gas. Yeah, jumping jack flash, it's a gas. Yeah, jumping jack flash is a fucking gas, man.
Is the real life? Is this just fantasy? Caught in a landslide, no escape from reality. Open your eyes, look up to the skies and see. I'm just a poor boy, I'm innocent, because I'm easy come, easy go, little high, little low. Anyway the wind blows doesn't really matter to me. To me. Mama, just kill a man. Put a gun against his head. Pull my trigger, now he's dead. Mama, life had just begun, but now I thought it throwing all away. Mama, didn't mean to make you cry. I'm not back again this time tomorrow. Carry on, and if nothing really matters. Too late, my time has come. Send shivers down my spine, body's aching all the time. Goodbye, everybody, I got to go.
We're back. So, continue the event. We will now present a video of Dr. Alencar Xavier. Unfortunately, he cannot be present, but he will try to participate in the round table in the finish the presentation. Okay. So Dr. Alencar Xavier got his PhD at Purdue University in soybean breeding and statistical genetics. Alencar has worked at Corteva AgriScience since 2016 as a quantitative geneticist and breeding analyst. And since 2017, he has been an adjunct professor at Purdue University. His research focuses on computational quantitative genetics, mixed model, Bayesian methods, and machine learning, and on integrating GBI, genomics, and the environmental information in plant breeding. So let's see your presentation. I share my screen.
Hello and welcome. My name is Alencar Xavier, and I'm here presenting today leveraging correlated information under multivariate settings. That is a presentation regarding the paper that I wrote with my colleague David Javier. A new approach fits multivariate genomic prediction models efficiently. Well, that's a brief outline of the presentation I'll be giving today. I'll start with a high-level rationale and start moving towards the statistical model that represents the problem we are trying to tackle. We are going to cover how we would be estimating coefficients and then how would be estimate the variance components. Coefficients and variances are the two main things we try to estimate when we are trying to fit a multivariate model. Later we are going to move into the simulations where we are going to present how our model compares to standard operations models, including RAML, for instance. Alright, so let's get started and jump straight into the rationale and statistical model. So, as you can imagine, many of us have the background in plant breeding, and one of the main objectives of a breeder is to improve the realized yield in the farmer's field. So in the analogy of the law of the minimum, when you have a barrel, the water will represent this yield that farmers realize in their farms. Of course, the amount of water in the barrel is always limited by the most limiting factor. Usually we think that those factors may include the yield potential, a resistance to a given stress, the stability of the material, diseases, and pest resistance. And as we identify these problems in our germplasm or in the lines, we try to address them through breeding such that we might be able to increase the yield farmers get. Well, we do operate upon multiple traits at the same time. And we are also aware that single trait models for genomic prediction in plant breeding are already well established. Some of those you might know as GBLUP or base B models, where you have one phenotype, series of genotypes, and you calibrate this model connecting genotypes and phenotypes. However, phenotypes may and do come from multiple locations, years, and quantitative traits. Many of those are genetically correlated, and one may consider using the genetic correlation among those traits to enhance the prediction, thus adding more information to every single prediction. So the rationale behind working with multiple correlated traits can be depicted here in a couple aspects. Well, we know that we can capture complex genotype by environment patterns if we decided to model the multiple environments as different traits, where they are considered correlated traits, but not necessarily the same. So we can also do the same if we perform this modeling with multiple phenotypes. And that usually translates in higher accuracy, because if you are jointly model correlated traits, you'll get extra power and more accurate estimators of breeding values. Second, we can assess phenotypic traits. We know that we are working in the era of phenomics. We generate data from multiple phenotypes, and the applicability of those phenotypes is measured by the genetic correlation between these high throughput traits and traits of interest, which could be things like yield, moisture, maturity, plant height, and disease scores. However, it's very nice to know that even though multiple trait models, multivariate models, multi-location models can be very beneficial from the theoretical standpoint, we also know those are computationally prohibitive. Why? Because every one trait you add into the model, you can increase the computational cost by the number of traits in exponential 7, according to Zoe Steffens 2014. So modeling multiple traits is not a common practice in the sense that it has not been a feasible technique to deploy. That being said, if multivariate models become more and more efficient over time, we know that there are plenty of applications that go beyond the genomic prediction per se. So here I'm providing two examples of what we can do with those genetic correlations, the multi-trait heritability values, and on the left I'm depicting two approaches of reduction of dimensionality that showcase and inform the trade-off among traits. So that's from a study that I did in soybeans a couple years ago. We have a principal component that is basically the eigendecomposition of the variance-covariance matrix of the genetic correlations. Well, more precisely the genetic correlations. So one thing that we see here, yield and reproductive length are aligned to the same vector. And on the right, what I have here is a graphical model also built upon the genetic correlation among traits, showing that yield is directly connected to three other traits: ACC, which corresponds to average canopy coverage, the number of nodes in the soybean plant, and the length of the reproductive period, here depicted by the abbreviation REP. So those are just a depiction of what you can do learning the trade-offs when you are feeding multiple traits. On the right, on another hand, we are doing a environmental classification where we treated different geographies as different traits. I ran the multivariate model, and I was able to create this map based on the genetic correlation among locations, which puts into perspective which locations have a better alignment in genetic terms when I'm breeding for different regions, and data from where could be leveraged where else. That can be also informative to determine or districting your target population of environments. That means that I can distribute my field trials differently, reallocate my resource differently based on my knowledge of this environmental classification that was purely based on genetic performance and how the genetic performance and ranking align from the different regions. I do not require for this analysis that all individuals be planted and observed in all locations. That's one of the beauties of multivariate models. They are connected through genetics, not necessarily through phenotypic records. So why would multivariate models be any better than the models we currently run by modeling one trait at a time? So here you can imagine that any phenotype described by the vector y could be a function of the genetics plus environment or genetic plus residuals. And the gain of information when this y is multiple traits is basically the correlation between trait 1 and trait 2. The variance of this joint vector y1 and y2 is depicted here, that the correlation we observe on the phenotype could be coming either from the genetic component, here depicted by the covariance A12, or by the residual covariance here, the sigma E12. So basically we are objectively adding more information into the model. Well, that looks a little high level, but I will show you in this slide here that we can actually compute where this gain is coming from. So from the same model shown in the previous slide, now I'm using matrix notation to showcase the same gain of information we would have using genomic information to estimate breeding values in a univariate model here below represented by G1. And on the very last line, what I depicted is conditional to the secondary trait, how we would be computing G1. And what I highlighted in yellow in this slide is objectively the information gain we get by modeling G1 conditional to G2. So even though it seems to be a straightforward solution, the fact that we are simply adding more traits, there is a whole chunk of the equation that is added when we are conditioning traits or fitting them jointly. Alright, so next, if you care to read the study we published, you see that we try to focus not on the GBLUP based on relationship, but instead using ridge regression, which is an equivalent framework. So regression type models are nice because if you store the marker effects, the beta coefficients, you can very nicely use them for predicting new data sets, as opposed to GBLUP, where you have to carry the estimation set with you every time that you want to predict newer individuals. Besides, ridge regression is easily compatible to multi-stage framework, where we use blues to adjust for the spatial variation. We take those blues, we connect into multi-environment BLUPs or the regress BLUPs, and we use those to fit linear models that include genomic information, which is a more computationally efficient way to handling the data. The third point here that I want to make is that ridge regression is a Gaussian model. It's fully additive depending on how you parameterize, but generally additive, and it is equivalent to GBLUP, and that's a known property of it. A big advantage of dealing with ridge regression as opposed to GBLUP is that we don't have to create or invert the G matrix. And that's an advantage. Why? Because G is not always inversible, and every time that you have to do some sort of bending or adding a value to the diagonal of G, you actually are choosing to lose precision. So the G matrix can be cumbersome, especially because we work in plants where more often than not we have more observations than markers. And last but not least, regardless of whether you use ridge regression or GBLUP, when you feed those models under the multivariate framework, you get this nice covariance and variance components that provide statistics that are very meaningful for breeders. That would include heritability, reliability, accuracy, genetic correlations. From those you can create selection indices, and with the covariance components you can also calculate the correlated response to selection. Alright, so how does this ridge regression model works in the multivariate framework? So the model looks pretty much like a univariate model would look like. You have your response vector of y, which corresponds to the phenotypes. We have the mu, which is the overall mean or intercept. Beta here corresponds to the marker effects. The vector E corresponds to the residuals. And the matrix Z is a block diagonal, where the diagonals have the genotypic matrices from the individuals from the various traits. Recall that you may or may not have any overlap of individuals between the different locations or the different traits. So why is that multivariate in any sense of the word? Well, because the covariance of beta, the marker effects, it's distributed as such that every time you update the marker effect for any given trait, it actually follows this covariance capital sigma beta that connects all the traits together. For this specific model we will be working today, we are going to assume that the residuals are independent, meaning that each trait will have its own residual variance. However, the residuals between traits will be independent, and that's a very valid assumption, because if you imagine that you're modeling individuals from various environments, there's no reason to believe that the residuals would be correlated by any reason. So you can imagine that if you will follow in the traditional multivariate framework this model we just described, the mixed model equation depicted in this slide can be enormous. And that's because every time we try to fit a model that is jointly modeling multiple traits, we have to operate using Kronecker products. That implies that the dimensions of the left and right side of our system of equations becomes really big. And another thing to keep in mind is that every time we compute or update the variance components, we have to invert the left-hand side of the equation. So just to illustrate, if we are working with maybe 3 traits and you have 1,000 markers, when we are working with univariate framework, the left-hand side would be 3 times 1,000 by 1,000. However, working with multivariate, you're going to have one 3,000 by 3,000 matrix. So I can safely say that as the number of observations and marker increase, fitting multivariate models, at least under the general sense or how it's usually done in the current framework, it's impossible. And here I want to just create the sense that it's a problem we're trying to address, we have been thinking over, and really computation is a resource that is still as expensive, even if we go through cloud computing. Modeling such large models may or may not converge, and it may take a large amount of resources. That will be time, that will be the cost of the computation, and you can imagine that we really need to get those values as soon as possible to make the breeding decisions. So here I just put it as unless, because drum rolls, we are going to really try to capitalize and focus on how to address this computational limitation in this presentation. All right, so let's move on to the coefficients. You may or may not know that every time we are feeding a model in that say apply to genomic prediction, we have more often than not more parameters than observations. So you can imagine that many models like the ridge regression blob, Bayes A, Bayes B, and all those Bayesian methods, we would be feeding a large number of markers, and more often than not more markers than observations. So it's a hyperdimensional model. The way how that is done is through a methodology called Gauss-Seidel. Why do we do that? Because handling such a large number of parameters does not justify creating the system of equations. So the first thing that I want you guys to keep in mind, we will be using Gauss-Seidel, which means that we will not be setting the mixed model equation as you probably have learned in quantitative genetics or the basic genetics class. So that's very nice because we don't have to do that x prime x, z prime z. Instead, the way how Gauss-Seidel works is by feeding one marker at a time, getting the residuals, and feeding the next marker. So here I'm depicting by a illustration where you can clearly see that the way how the model is updated is by feeding one or a few markers at a time, and you do that over and over and over, and the model eventually will converge to the exact same solution as it would if you were setting the system of equations. How that look like in the multivariate setup? Well, the formulation is rather similar, except that when we update a marker, we update that marker effect not as a single value, but as a vector, because the marker effects associated to the multiple traits now, you can imagine that instead of feeding that one marker for one trait, you're feeding that marker for several traits. So if you have 5 traits, beta will not be a single value, but instead a small vector of length 5. So that means that it will be running the Gauss-Seidel in a small system of equations 5 by 5. It is yet possible to notice that even though we are now feeding this small system of equations that has the same dimensions of the number of traits, we can go a step further and use conditional expectations and fit one by one using vectorial mathematics. But the whole idea that I want to really convey here is that the formulation we use in the multivariate system of equations using Gauss-Seidel is really small. Instead of fitting those huge markers involving large chronic products, instead, by fitting one marker at a time, we are handling very small matrices that for the simple example here is just number of traits by the number of traits. So the idea is still as feed one marker at a time, take the residuals, feed the next marker. And the nicest thing, we are avoiding that big chronic product altogether, and we are not doing any matrix inversion, and still we are getting all the advantages of a multivariate model. So one of the only matrix that we actually have to invert, and that's only once every full iteration cycle, is the variance covariance matrix of marker effects, here depicted by capital sigma beta. So that here I'm just further depicting how that loop would look like. So basically I'm color coding on what do we compute once, what do we compute once every iteration, and what we compute in each marker within each iteration. So in green are things that we compute before even starting the loop. So the zeta prime zeta for each trait is computed only once before the loop even starts. In orange are the marker effects and zeta prime error, which we do compute for every marker within each iteration. And in blue here are the variance components, which we compute once every iteration, but not for every marker. So we just compute it once in each iteration. And if you recall, one of my first slides where the covariance was the information being added into the system of equations, that is where most of the benefits come from. So here in this bivariate example, we can clearly see that the sigma beta occupies those off-diagonals that correspond to the covariance between traits. Another thing that we point out in our study is a rather new discovery that corresponds to the fact that if you update the markers in random order, you can truly speed up the convergence. So on the left here, we are updating the marker effects in random order using the randomized Gauss-Seidel, and the whole mixed model equation with 10 traits and 5,000 observations and 4,000 markers converged within 50 iterations, probably a 20-30 seconds operation. The same model with the same data if we update the marker effects using ordered Gauss-Seidel, meaning a loop in the same sequence of markers. It took as many as 3,000 iterations. Well, to be fair, 3,000 is still much less than what you'd have to do if you were to do Monte Carlo. If you were to fit your model using MCMC, which is between 30 and 100,000 iterations. But we can reduce dramatically this number of iterations from 3,000 to 50 just by updating the markers or marker effects in random order. In fact, the randomized Gauss-Seidel is something that came from the machine learning literature as opposed to the classical quantitative genetics statistics literature. Alright, so now that we know how to update the marker effects, you may be wondering where do we get those variance components from, right? So I'll briefly show how where we're getting those variance components in the univariate and the multivariate case. First, a very brief history of variance components. The whole variance terminology came from Fisher in 1915, but prior to that there were analyses that were not yet named ANOVA, but they depicted the same principles of that type of analogy. So in the very top, I'm mentioning this reference of Airy, 1861. The theory of ANOVA is formalized in 1921. Keep in mind, ANOVA is a nice framework, but it has some very strong assumptions. It requires balanced data, and it can hardly handle complex covariance structures. 1953, Henderson published a manuscript where he generalized ANOVA to an infinite number of fixed and random effects. So one of the breakthroughs was starting introducing matrix notation within the variance components literature, which has been a limiting factor. So that was 1953. 1962, the idea of a restricted likelihood is becoming more and more mature, and the term RAML was proposed. From that point, you can imagine that computation was a very limiting factor, and there were two possible paths to which Henderson methods and restricted likelihood methods would pursue. So on the right here, I'm depicting the exact likelihood path, which started with that idea of estimating the maximum likelihood function, which could be maximized at a time was proposed to start using things like the expectation maximization algorithm in case there was no exact solution. And at a lighter computational cost on the left, I'm illustrating methods that were iterative at a time. So some of the first iterative methods that became quite popular were proposed by Cunningham and Henderson in 1968. The same method was later corrected and generalized by Robin Thompson, to accommodate non-orthogonal design matrices. Henderson kept working in iterative methods. So one of the methods that became also quite popular was called the Henderson simple method from 1980, and some iterative variations that he proposed with his student Van Vliet. Going back to the Remo path, as this idea of likelihood and quadratic expectations evolved, there was part of the literature that branched out to MIFQ and MinQ frameworks. Those were largely the contributions of Rao. Very nice framework, but with a very dense matrix notation. At the same time, Patterson and Thompson, they were working towards providing more and more insights, making Remo a more clear approach to pursue. So in later years, the methodologies by which we estimate variance components in our plant and animal models was established by Henderson, and it had plenty of iterations and intermediate solutions. Some of the solutions that became really famous were through derivation-free Remo, where we use numerical methods and assess the likelihood. And one of the most modern implementations of Remo is the average information from Johnson and Thompson from 1994, which is implemented in software such as ASRemo, BLUPF90, and others. However, there was this one branch that I left for last that I call the fast variations. So we know that Remo became the gold standard, but there were also fast variations that were not necessarily Remo, but they were unbiased, they were inexpensive to compute, and they have similar properties as Remo, though they don't share all the exact same properties. They were, invariant, center invariant, unbiased, but for a reason or another, people did not pursue any further. So two of those methods were the pseudo expectation proposed by Schaeffer in 1986, and the tilde hat method proposed by Van Reyden and Jung in 1987. This one, the tilde hat, is more often than not referred to as a Van Reyden method. So the nice thing about those methods is that at a time, the authors, both Larry Schaeffer and Paul Van Reyden, were analyzing data sets with millions of individuals in late 80s. So they have plenty of data. They were using the animal model to calculate blubs of breeding values and really working in the theory of animal breeding. However, they didn't have the computational power to estimate the exact Remo solution. So they proposed these creative ways of calculating variance components. And once again, many of those methods were created to handle very large data sets. and for a reason or another, they were forgotten as we move into the genomic era. So here I'm just briefly describing how it came to be this variance component methods. So on the very top we have the first derivative of RAML, we have that restricted likelihood, and then we do the first derivative with respect to the sigma squared beta. And on the top we have the covariance between y and u, which is ypv py. Here are all the definitions of the P matrix, which is the null space projection, divided by the trace of PV. Oh, very nice. You could get the RAML solutions through this derivation, but that would entail that you need this V inverse matrix and matrices such as the covariance of K. So in 1986, Schaeffer proposed this method where he said, well, maybe if we get rid of V altogether, we can come out with a very simple expectation where, keep in mind, whenever you see traces, we don't actually compute the trace. We don't have to build the Z prime Z, we just compute directly the diagonal elements, so we never get to do the Z prime Z. So that was very nice because this type of framework is directly compatible to the things we have to generate to solve the Gauss cycle. And then in a year later, Van Reyden proposed that maybe we can do a slightly better job, not by getting rid of the V inverse matrix, but by replacing by the inverse of the diagonal elements, which is much simpler to compute and yet can preserve some better statistical properties and keep it closer to the RAML solution. Besides, if your model is close to orthogonal or unstructured, the tilde hat will likely give you exactly the same solution as RAML. The name of the method is called the tilde hat because he approximates one side of the equation by calling the y d minus 1z as b tilde. So both methods are equivalent as long as the if the D matrix is just an identity matrix, the tilde hat is reduced to the Schaeffer method, pseudo expectation. So why do we care only about the genetic covariance? Well, because the residual covariance under RAML or any other method is really straightforward to be computed. So the really hard variance component to get is the genetic variance covariance. Plus, not having to do V, we are, it's a very painful matrix to compute. It takes a lot of memory, so we do not want that. And notice that here we are not using anything with the C matrix, which is the left-hand side of the mixed model equation either. So basically, what do we keep in memory, or what's the solution of those variance components? It's rather straightforward. It's just the B tilde times the prime B hat divided by the trace. One more time, we don't have to calculate Z prime Z in the inverse, we just use the diagonal elements to get a trace. Same with the covariance, it's just the tilde hat from trait 1 and 2 plus the hat and tilde of traits 1 and 2. We get the same solution as Schaeffer by just assuming that D is an identity matrix. We have very few things stored into memory, just the B hat, the B tilde, and some of the diagonals of ZPZ. The beauty of these algorithms is that we do not have to keep in memory the variance covariance matrix. We don't have to keep in memory the left-hand side equation of the Anderson. We don't have to calculate determinants, and we don't have to do any dense inversions at all. A more intuitive derivation of the Schaeffer method, it would be simply getting the corrected values or centered values of trait A being predicted by B, and the B being times the prediction of B from A. So a intuitive interpretation of genetic covariances would be how well A predicts B plus B predicts A, and you take somehow like an average of it. So it's a very intuitive derivation being projected in mathematical terms. What do we do with all this variance components? Again, it's very nice. We can get the genetic variance, we can get the heritability, and we can get genetic correlation among traits. So those are all very informative genetic parameters, very applicable to plant breeding, and you can solve all of those very efficiently. So the model not only gives you the marker effects, but gives you the components you need to calculate heritability, variance components, and genetic correlations. So let's go now to the factual benefits and the comparison between the proposed method and the gold standard. The metrics we have been evaluating for this study include the computational efficiency, which is basically how long it takes to fit the model. When evaluating the breeding values, we're going to check the accuracy, where that is defined by the correlation between GBVs and true breeding values. And last but not least, we want to look at bias and precision of our coefficients, where bias is the expectation of the difference between our estimator and the true value, and the precision corresponds to the standard error or standard deviation of this measurement. We're going to look into two scenarios from two data sets that are publicly available. One is a wheat data set available in the BGLR package, and the other one is a soybean data set available in the SoyNum package in R. In the first scenario, we're going to use the genomic available in the true data set and simulate phenotypes coming from 10 environments where all individuals have been observed in all environments. However, so we are calling this small data set the balanced data set because everyone has been observed everywhere. The second one is called the unbalanced scenario because we are using the true genomic data but simulating phenotypes from 10 environments. Each individual will be observed in a single environment, and the number of lines per environment is 514. So we just observe 10% of the data set in each environment, and we're going to fit the 10 environments being treated as 10 different traits. And the number of markers is slightly different. In one case we have 1200, and in the soy data set 4300. One thing that I have to disclose is that under the unbalanced scenario in the soybean data set, we were not able to get RAMO genetic covariances. Whenever we had zero overlap of records, the estimated values from RAMO are all anchored at zero. We tried that in ASRAMO, RAMO F90, AI-RAMO F90, and a couple other software. And in no implementation we are able to get meaningful variance covariance components from RAMO. Once RAMO has some overlap of individuals, the estimates of variance components became available, but in that soybean data set we were not able to get any, so we will not be, unfortunately, benchmarking against RAMO in one of our scenarios. Simply because RAMO could not be used in such an unbalanced scenario. Alright, so let's look at the how long it took to fit the model into the small data set that was balanced. Here we use a couple of different softwares. The methods we proposed, we named them PEGS and THGS, meaning pseudo expectation Gauss-Seidel and tilde hat Gauss-Seidel. The software we compared against were ASREMO 4.2. That took 272 seconds. AI-REMO F90 took 100 seconds. And the Gibbs F90 took 560 seconds. The proposed methods that we discussed in this presentation took less than a second, so 0.27 for the multivariate case, and we also fit the univariate tilde hat Gauss-Seidel one trait at a time, and we added them up, and that added up to 0.23 seconds. For the second scenario, where we do not unfortunately have REMO, both PEGS and THGS gave roughly the same elapsed time, so we don't have to compare one against the other. But what we can do is to increase the number of traits to see how long that will take to feed the model. So on the left we are checking what would happen if we were to feed that model with 4,000 markers or 42,000 markers, where the number of traits is 10, 50, or 200. And here's the elapsed time. So to fit 10 traits, it took 0.2 minutes. So, less than a minute. To 50 traits, it took us 3 minutes. And notice that even if the number of markers is really high, 10 traits is still under a minute, 50 traits, roughly 10 minutes. And then with 200 traits, it took 80 minutes, so a little over an hour. And with 200 traits and a large number of markers, it took 2 hours. Still, that number of traits and markers would be rather inconceivable to run with a REMO solver. Now let's look at the accuracy. So, because our phenotypic records are simulated, we did have the true breeding value, and we could directly assess what's the impact in accuracy by using REMO or the different methods. So on the left, we are also trying here the TVGS, which is the true value of various components fitted with Gauss-Seidel. So that was always the very best because we were using the true variance components. Variance components estimated through REMO were equivalent to the tilde hat and pseudo expectation. So the accuracy with our proposed methods really capture the same covariance among traits that you'd get from REMO. So that's fantastic. And what you can see is that the univariate methods falls behind because it cannot leverage that information across traits. The general trend is that high genetic correlation and low heritability favors multivariate methods over univariate methods. But besides that, all multivariate methods provided very similar accuracies. Of course, using the true values for the variance covariance was the very best. Then REMO, THGS, and PEGS. Now let's look at the bias of the heritability. So the largest discrepancy we saw in the comparing REMO to the proposed methods was 0.05. That happened when the heritability was really high and the genetic correlations were low. So the proposed methods, they underestimated the heritability of the trait by on average 0.05. Now the bias of the genetic correlations. At full disclosure, genetic correlations are things that are really hard to estimate in general. It's hard for REMO and it's hard for all the methods. A very interesting thing here is that when we had high correlation and low heritability, the difference is 0.07, not because our method was bad, but because REMO was unable to do a good job. So under those circumstances, Ramo, not our method, underestimated the genetic correlations by 0.08. And then in the next scenario, Ramo overestimated when the heritability was 0.5 in high correlations. Ramo overestimated the correlation among traits by 0.07. So another thing to notice, the soybean data set is larger in the sense that it has more individuals and more markers, and the heritability and genetic correlations are largely unbiased. Now precision. So precision is the standard error of those heritability and genetic correlations. So let's start with the precision of the heritability. So it's true the pseudo expectation until the hat had a standard error larger than as the ones of Ramo. So something had to give, and one thing that our methods is a little worse than Ramo is the precision of the heritability and genetic correlations. Similar trend here now depicted in the genetic correlation is that our methods, especially when the correlation among traits was low, it had a larger standard error than Ramo. Here, on average, 0.07 larger. But fear not, our method was actually meant to be used in larger data sets where it's not possible to fit Ramo. So as the amount of data increases, you get less bias and more precision. So here we ran a quick comparison using PEGS, THGS in the univariate THGS with either 250 observations per environment or 3,000. So in the soybean data set, we had as many as 5,000 individuals, so it was fine to use up to 3,000. It was not even half of the data set. So the accuracy has a tremendous improvement as you increase the number of observations. That is true for univariate and multivariate settings, more so for the multivariate. The slope or the bias of breeding values, as we got to the 3,000 observations point mark, it went from slightly biased downwards, 0.98 to 1, so it became fully unbiased. The bias of heritability was very small, 0.01, not significantly different than 0, and even 0 in the THGS case. But overall, I would say that slightly underestimated, but not different than 0. The standard error of the heritability dropped from that 0.07 we were looking at earlier to 0.04, and presumably, which was the same as the Ramo report. But presumably, as the number of observations increase, your standard error, your precision will increase, standard error will decrease. Same thing with genetic correlations. It was slightly biased for the 250 observations per environment. They became fully unbiased at the 3,000 individuals per environment mark. And likewise, the precision increased by which is measured by the decrease in standard error. So the standard error of genetic correlations went from 0.17 to 0.13. So limitations and other things to consider. For the simple implementation we made available and we described in this manuscript, we just use a method where the intercept is the only fixed effect, and that's when the methodology works at its fastest. That being said, having more and more random effects does not increase by a lot the computational cost of our method. It just means that we have to run an absorption of the fixed effects prior to run the model, so that it may increase by a few seconds, but that is regardless of the number of markers and observations. The correlated residuals. So one of the things that we got away and really increased the speed of our method was to avoid the Cronacter product But if the residuals were correlated, we will not be able to get rid of this Kronecker product. And you might be thinking next for kernels and SVD. What happens if I have millions of markers? So one solution is to create a GBLUP and eigendecompose the genomic relationship matrix. And that can be done such that if you run a ridge regression of the principal components, you get the same result as running a GBLUP or a ridge regression. So that's an efficient way of bypassing the problem of having millions of markers to estimate. Another thing that's very interesting is that as the number of traits increase, and here we're talking 500 plus traits and traits that have very high correlation among themselves, it might be necessary to bend the covariance matrix here depicted by sigma beta. Bending simply means that we shrink a little bit off diagonals. And one thing that's nice to mention is that if you have up to 10 or 20 traits and your data is perfectly balanced, it is possible to get a RAMO to be efficient as well through a approach called canonical transformation or kernel diagonalization. So there are cases where RAMO can be efficient as long as your data is perfectly balanced and the number of traits is not too large. A general recommendation for multivariate models, as you are looking for estimate those covariance components and get that extra predictive ability, is that if you have very small data sets and you feel comfortable with RAMO, just go for it. RAMO is a little cumbersome to handle, but if your data set is small, you might as well just use RAMO. In that range of 5 to 20 traits for a small to moderate size data set, people have been using the past Gibbs sampling, which is more stable than RAMO, with very satisfying outcomes. However, if your number of traits is large or your data set is large, we would recommend you to move to the proposed approaches, PEGS, THGS, so pseudo-expectation Gauss-Seidel or tilde-hat Gauss-Seidel, which can really speed up the computation of large models. And myself, I'm very comfortable with running those models even if the number of traits is not too large. And last but not least, a model that is somewhat novel, just like the methods we are describing in this study, is a approach developed at the UC Davis by Dan Runcie called the MEGA LMM, which is a model for highly correlated traits, and when the number of traits is larger than the number of observations and number of markers. So if you have thousands and thousands of traits in a relatively moderate size, small to moderate size data set, you can run this MEGA LMM, which is it's an MCMC based method, but with a very efficient decomposition and works reasonably well for a very large number of traits. So the conclusion, just for the final remarks, is that multivariate models are very valuable. We need them to get those genetic correlations, that you can do a whole lot, classify traits, classify environments, and create selection indices. But in the past, they have been computationally unfeasible. The second remark is that now with this efficient estimation of coefficients through the randomized Gauss-Seidel and efficient variance component estimation through pseudo-expectation and tilde-hat, we can run these very large multivariate ridge regression models. And last, we compared those methods. We showed that the tilde-hat and pseudo-expectation are very suitable replacements to RAMO, especially for large data sets. And with that, I thank you all, and I hope I didn't bore you too much with the mathematics, but I hope you understand that this methodology is very valuable, and as we understand more and more the usage of multivariate methods, that can be a big enabler enabling technology in plant breeding. Thank you very much.
Mara, I'm so sorry, but your mic is off.
Sorry, guys. Obrigada, Jean. Pessoal, vamos dar início agora à mesa redonda. Eu vou conduzir em português mesmo, tá bom? Como vocês viram, o Dr. Alt, ele não estava muito bem, ele estava com a gripe e ele não pôde ficar para esse momento. Mas tanto as perguntas que vocês tiverem, né, alguma dúvida sobre a apresentação do Dr. Alt e também do Dr. Alencar Xavier, vocês podem estar entrando em contato com a gente ou diretamente, né, no e-mail deles. O Alencar Xavier, ele está participando de uma banca de tese e foi por esse motivo que ele nos enviou a apresentação. Infelizmente, ele não conseguiu chegar a tempo para participar da mesa redonda, então vou conduzir só em português mesmo com o Dr. Ricardo. E eu convido então o Dr. Ricardo para a gente dar início à sessão de perguntas aí da galera que mandou aqui no chat. Tudo bem? Eu tenho uma pergunta, eu acho que já posso começar com a minha, pode ser? Dr. Ricardo, parabéns pela palestra, excelente. Não é muito a minha área, eu não trabalho com esses dados voltados para Machine Learning. O nosso laboratório, a nossa equipe está começando agora a trabalhar, né, nesse sentido. E você comentou que para a criação de uma árvore de decisão, é necessário ter uma característica com alto ganho de informação e precisa ter a questão da entropia, deve ser baixa, né? Então, a característica tem que ter esse balanço. Tá certo isso?
Quando você escolhe um atributo para colocar na árvore baseado no ganho de informação. Né? Esse ganho de informação é a minimização da entropia.
Então, tem uma forma de tentar diminuir essa entropia? Qual que é a melhor forma para diminuir e aumentar esse ganho de informação?
Então, a minimização dessa entropia é buscar por um atributo que divida melhor o seu conjunto de dados.
Entendi.
Quanto mais você produzir subconjuntos que são puros, de acordo com alguma característica que você quer, seja ela categórica ou numérica, menor vai ser a sua entropia. Consequentemente, maior vai ser o ganho de informação, de acordo com essa estatística que a gente viu, esse cálculo, né? Mas existem outros cálculos, existem outros índices, como o índice Gini, por exemplo.
Entendi. Então, algumas características que são mais complexas, elas são mais difíceis para atender a esse balanço, a essa diminuir, no caso, ter uma baixa entropia? Como que funciona com características mais complexas, por exemplo, com índices de vegetação, bandas espectrais?
Então, não sei dizer exatamente, dependendo não sei, assim, para essas características específicas que você disse, eu não sei. O que eu sei dizer é que, assim, quanto mais complexo é o conjunto de dados, né, então, consequentemente, imagino mais complexa é a característica que você está investigando. Então, vou dar, no nosso caso aqui, por exemplo, característica é a produtividade de arroz, ou teor de amilose de arroz, ou dureza do grão, porcentagem de inteiros, né? Então, dependendo dessa característica, seu dado vai ser ou mais ou menos complexo. Ele sendo mais ou menos complexo, né? Aí vamos falar dos SNPs, né? Ele sendo mais ou menos complexo, os SNPs vão explicar mais ou menos aquela característica que você está buscando. Se a explicabilidade for menor, isso vai resultar provavelmente numa entropia maior. Então vai depender tudo disso, né? Mas essa análise estatística você faz por variável, né? Não tem para dizer não tem como dizer antemão qual, assim, como que vai ser a entropia do dado. Né?
Entendi. Tá bom, muito obrigada. Vamos então iniciar as perguntas que os participantes enviaram no chat. Essa aqui é do Jean, ele parabeniza pela excelente palestra e ele gostaria de saber, né, ele trabalha com seleção de genótipos de milho pipoca com tolerância à seca. E ele gostaria de aplicar, né, o uso do machine learning no experimento dele. Então, ele queria saber quais que são os primeiros passos para fazer isso.
A primeira coisa que eu faria...
Só um minutinho, continuando. Ele poderia usar ferramentas de base simples de dados ou apenas de um conjunto grande de dados? Qual que seria mais eficiente? E se a decisão para utilizar o machine learning poderia ser feito após o término do experimento?
Então, deixa eu ver se eu consigo entender. Acho que a primeira coisa que você tem que fazer num conjunto de dados é uma análise exploratória dos dados, né? Para ver quais as características de cada atributo que você tem, certo? A distribuição das variáveis, né? Se elas estão dentro de uma normalidade ou não. A primeira coisa que eu faria, né? E depois, a escolha do algoritmo que vai utilizar depende muito do que você quer para a sua aplicação, né? Então, se você quer, por exemplo, uma maior taxa, vamos supor que você, né, que dentro da sua aplicação você busca uma taxa de acerto, seja uma análise preditiva. Então, se a sua prioridade seria essa maior taxa de acerto, pode ser melhor trabalhar com modelos que eles não priorizam a interpretabilidade em busca de uma maior taxa de acerto, como, por exemplo, redes neurais artificiais, né? Agora, se o seu objetivo é ter uma maior interpretabilidade, por exemplo, mas sacrificando um pouquinho de taxa de acerto, então eu poderia ir para uma árvore de decisão, como eu mostrei, né? Com relação à quantidade de dados, quanto menos dados você tem, menos hiperparâmetro, menor, vamos dizer assim, menos complexo, na teoria, deve ser o seu modelo. Porque quando você tem poucos dados, há uma tendência ao seu algoritmo de aprendizado de máquina ele overfitar, né? Há um overfit em cima dos seus dados. Então, vamos pegar como exemplo redes neurais artificiais, que eu acho que até falaram, se não me engano, numa palestra de um professor anteriormente, que ele deu que tinha uma imagem lá de um modelo de redes neurais, né? Quanto maior o número de pesos, maior essa sua rede neural, né, mais há uma tendência dela decorar os dados que você está utilizando, né? Então, é o que a gente chama, vai ter uma variância muito maior em cima dos seus dados e tem uma tendência a decorar. E quanto maior, então, nesse caso, você vai precisar de mais dados. Então, assim, se você tem poucos dados, a sugestão é para tentar um modelo mais simples primeiro, e não um modelo que envolve tantos hiperparâmetros, certo? Então, respondendo essa pergunta, você pode usar essa ferramenta em dados pequenos ou somente em Big Data? Não, pode usar em dados pequenos, pode usar em conjuntos de dados pequenos. Mas, neste caso, utilizando algumas técnicas para você diminuir o número de hiperparâmetros que a gente vai ajustar do modelo, tipo, né? Então, por exemplo, no caso de redes neurais artificiais, você tem que trabalhar com redes menores, com menos pesos para ajustar.
Show, muito obrigada. Eu acredito que respondeu. Aí, Jean, ficou a dica, então. Vamos para a próxima. Essa é do Charleston Lima. Excelente palestra, Dr. Ricardo. Existe um número mínimo de dados para aplicar o método, o modelo?
Então, essa é uma pergunta também que, assim, eu não conheço nenhuma relação analítica, assim, entendeu? Um número mínimo de exemplos para para que haja aprendizado. A teoria do aprendizado estatístico, que ela diz que dependendo do número de exemplos muito pequenos, não ocorre aprendizado, né? Obviamente, se pegar um conjunto de dados, aí você tem 10 amostras, né? É muito difícil ocorrer aprendizado, certo? Agora, se você tiver 150 amostras, por exemplo, sendo que elas estão extremamente muito bem distribuídas, vamos supor que seja um problema de classificação, novamente, uma tarefa preditiva. Você tem 150 amostras, por exemplo, divididas em 3 classes, 50 em cada classe, extremamente bem divididas, entendeu? Num espaço, né, de variáveis. Então, nesse caso, pode ocorrer aprendizado. Agora, se você tem, sei lá, por exemplo, 5, 10 exemplos por classe, vamos supor que seja uma tarefa preditiva novamente, aí é muito difícil correr aprendizado. Mas eu não sei dizer assim, uma relação analítica, né, de qual é o mínimo de dados para que haja aprendizado. Né? Mas, assim, em teoria a gente diz que quanto mais dados, melhor.
Ok, muito obrigada, Dr. Ricardo. Temos mais perguntas? Eu acho que essa foi a última. Deixa eu ver se tem algum aqui no chat privado. Não temos perguntas. Dr. Ricardo, gostaria de agradecer a sua participação. Infelizmente, a round table não deu para ocorrer, devido aí aos outros compromissos dos palestrantes, mas agradeço muito você ter ficado até o final e ter aceitado o nosso convite também. Muito obrigada.
Não, eu que agradeço, estou sempre à disposição. Espero que vocês tenham, que tenha sido útil a palestra, que vocês tenham adquirido conhecimento.
Com certeza, foi sim.
E estou sempre à disposição. Podem mandar e-mail, nosso grupo está sempre aberto.
Muito obrigada. Pessoal, chegamos então ao final do nosso primeiro dia de evento. Eu gostaria de agradecer a todos vocês e todos os participantes que compareceram e ficaram com a gente nessa tarde maravilhosa. E eu gostaria também de agradecer aos nossos apoiadores e patrocinadores, a Corteva AgriScience, a Infoline, Banda Larga, a Rio Norte Sementes, o Grupo Conecta Gen e a toda a comissão organizadora do Grupo InterGen. E eu convido todos vocês para continuarem nos acompanhando amanhã. O segundo dia do evento terá como tema central New Genotype Technologies, Main Result and Contribution. Então, fiquem bem e vejo vocês amanhã. Até mais. Tchau, tchau.